Sajasna madam.
Sir.
Madam, it is audible now?
Yes, sir, it is audible. My voice, sir?
Yes, madam, it is audible, madam. I think control, you can mute yourself, all those things you can do, madam, yourself itself.
Okay.
Control will be available. In
Okay.
case any problem means, you can tell me, madam.
Okay, sir.
Madam, that remaining peoples also join within your ID only,
Yes, sir. Here, Preeti madam came, but her session is in the afternoon, sir.
Okay, madam.
The madam not came.
Vishaka madam is in upside.
Vishaka madam is there here.
Okay, madam. So you all people in library only, na? So, hello?
Library only, sir.
Okay, madam. If any help means, you can call that G. Ravi sir, Murali sir. So they will be
Is also here only, sir. Here only.
Madam?
Murli sir is here only, sir.
Okay, madam. If any help means, you can ask them, madam.
Okay, sir. My voice is audible, no?
Audible only, madam. Dr. Sasank, sir.
Good morning. Can you hear me?
Yes, sir. Yes, sir.
Yeah.
I want to know your user ID. That's why, sir. Okay, sir. Good morning, sir.
Yeah. Good morning. Good morning, sir.
Good morning, Dr. Sasank.
Yeah. After a long break, I can see you now.
Yes, yes. Welcome to MNR College of Pharmacy and the International E-Conference.
Thank you.
Just we'll have a 15 minutes inauguration, followed by we'll have your lecture.
Yeah, sure. No problem.
Okay. Dr. Saravanan, we can start, sir.
Okay, sir. Sai Josna, madam?
Yes, sir.
Yes, madam. We can start program.
A very good morning to one and all. I myself, Sai Josna, Assistant Professor. On behalf of MNR College of Pharmacy, I happily welcome all the guests, faculty, students, and delegates from different pharma fields to the International E-Conference on recent developments in biopharmaceutical sciences. MNR College of Pharmacy is one among the 73 top grade institutions of MNR Educational Trust. This trust was formed in 1974 by Sri M.N. Raju Garu with the noble aim to shape the students into proud citizens of this nation. Sri Ravi Verma Mantina, the dynamic vice chairman, adds a perfect blend of modern approaches to achieve this goal. And the MNR College of Pharmacy is established in 2004. It is approved by AICTE and PCI, and it is affiliated to Usmania University, Hyderabad. And the different courses offered by MNR College of Pharmacy are B.Pharm, PharmD, M.Pharm, and PharmD Post-Baccalaureate. The college is having state-of-art pharmaceutical laboratories, and the library is having more than 1,000 volumes of specialized books and subscription of journals, and even the digital library facilities too. And the college is providing the co-curricular activities like these conferences, lectures, and industrial visits other than the curricular activities. And the college is having more than 50 faculty members. In that, the 20 faculty members are with PhD. And the college is running under the great guidance of Professor V. Alagusamy sir as a principal, who is rendering his valuable service.
Madam Sai Josna, madam, please unmute, madam. Madam, please unmute yourself and talk, madam.
Yes. Sir is having a vast experience with more than 23 years of teaching and research experience. He is a recipient of Young Scientist Award and member of AICTE, Board of Studies OU, and many more. Sir is having more than 150 plus research papers, and he is the editor of one international journal too. And sir is having 6 books and 1 patent to his credit. And recently, sir has been awarded as top 2% scientist in the world by the Stanford University, USA. I heartfully congratulate Dr. V. Alagusamy sir on behalf of all the staff members and MNR educational family for achieving this great thing. Every event begin with this short introduction. We'll start the program. Once again, I welcome you all for this e-conference. Every event begins with a prayer song. So it is our tradition to start this event with a prayer song. So let us begin the event with the prayer song.
Shri माच्युत शंकर प्रभुति देवै सदा वन्दिता क्षमा पातु सरस्वती भगवती
Thank you, sir. Now I request our principal, Dr. V. Aligarsamy, sir, for the inaugural address. Please, sir.
Good morning, all the delegates from various parts of the globe. On behalf of our management, Sri M.N. Raju Garu, the chairman of MNR Educational Trust, the vice chairman, Sri Ravi Verma Garu, all faculty members of MNR College of Pharmacy, I welcome all the delegates and the resource persons of this today's August event, which is happening with the support of all the pharma fraternity. MNR College of Pharmacy conducts every year a national conference with 1,000 delegates. This year, due to this COVID-19 pandemic, we could not conduct this national conference. But we wanted to take this as an advantage. Hence, we have planned this international e-conference on recent developments in biopharmaceutical sciences. We coined this term for this today's conference because the pharmacy is the significant place in the healthcare system in order to heal any of the disease. Today, the whole world is pandemic due to COVID-19. Here, biotechnology or microbiology plays a very important role in the healthcare system. Hence, we have chosen this topic of biopharmaceutical sciences and its advances in this field. To learn this topic from various parts of the globe, we have invited the resource persons from different institutions and research organizations. All the participants and all resource persons came forward on their own interest and accepted our invitation, all the resource persons without any hesitations. So we had such eminence in the field and who are agreed to deliver this today's speech. It's our fortunate for all the delegates, including me, to have such a wonderful resource persons in this August gathering. Let's all thank for such a forwarding or such an enthusiastic mindset to deliver to the international students. We shall thank to all the resource persons who came forward to deliver this lecture, and we shall try to benefit from them because they are spending their valuable time with us without expecting any returns from us. The returns what we are giving for them is the try to understand and try to do the research like them, because they are all one of the distinguished in their each of their field. Importantly, we have got the speakers from the different countries in order to have this international conference as a versatile with the resource persons, so that each one and enjoy this today's conference. I hope this in this platform that is provided by our management, let us make use for the scientific developments and benefit for the scientific community. We, the pharmacists and all the biotechnologists, try to contribute to the suffering humanity. With this motto, let us start this conference. with the first speaker, I think Dr. Sasank Sethi, who is from the Stanford University. So I welcome Dr. Sasank, and our the speaker will be introduced with his background by the coming event. So thank you, Dr. Sai Josna, for giving this chance to address this gathering in this wonderful occasion. Thank you, one and all. Let us have this August day as a memorable one in this scientific field. Thank you very much.
Thank you, sir. Thank you so much for the inaugural address. Now I request Dr. G. Sharavanan, sir, Professor, Department of Pharmaceutical Chemistry, to introduce our first session speaker. Please, sir.
Yes, very good morning to one and all. So before I introduce the speaker, that our participants to that Zoom ID was already reached on a 300, and so many people are trying. So we got already live streamed that the programs in on a YouTube. That YouTube link was already sent to your registered email ID. So please instruct your friends, those who are unable to join in the Zoom, to go to that YouTube link and watch that program in on a live. So very good morning to one and all once again. So it is a great pleasure for me to introduce the today's speaker of that first session, Dr. Sasank Sethi, School of Medicine, Canary Center at Stanford University, USA. Sir completed his PhD in the year 2020 from Pondicherry University in Nanoscience and Technology. In 2014, in the same university, Sir completed his MTech and he got on a gold medal in that MTech. In 2012, from Anna University, Sir completed his BTech in biotechnology. Dr. Sasank Sethi's areas of research interest is biological effects of therapeutic ultrasound on mesenchymal stem cells and impaired organs for regenerative therapy. Sir have around 10 international journal publications. The most important thing is the average impact factor of publication is around 6. And we people know how much difficult that we want to publish in an impact factor 1 and 2, but the SARS impact factor, average impact factor is 6. So that it shows that how much SAR is having impact on that research. SAR is a life member of American Chemical Society, USA, Royal Society of Chemistry, USA, Material Research Society, USA, International Society of Optics and Photonics, USA, and SAR is also a life member of Indian Society for Technical Education, India. SAR is having many honors and awards for his credit. Among that, here I was mentioned few of them. In 2018, SAR got an Inspire Senior Research Fellowship Award from DST. From TIFR, in 2017, he got SABIC Young Presentation Award. From 2017, from CSIR, SAR got Senior Research Fellowship Award. And from DST again in 2016, SAR got Inspire Junior Research Fellowship Award also. 2 times SAR got Travel Grant Award from DST SERB. In the 4 times in the year 2009, 11, 16 and 19, Dr. Sasank Sethi got Best Paper Presentation Award in various international conferences. And in various workshops and training, SAR becomes the place in the top 25 participant in the year 2015 and 2017. SAR also delivered plenary lectures in various national and international conferences. Currently, SAR is working as a postdoctoral researcher in School of Medicine, Canary Center at Stanford University, USA. With this brief introduction, I am very happy to welcome Dr. Sasank Sethi for this international e-conference on recent developments in biopharmaceutical sciences. Sir, please welcome and please deliver your lecture on nanotechnology, a new dimension for stem cell therapeutics and diagnostics.
Dr. Sasank?
I'm unmuting, sir. Just a minute. Sir, please unmute yourself, sir. Dr. Sasank, sir.
Yeah. Thank you for the brief introduction. So before I start my presentation, I'll just like to share my screen. I think Dr. Sarvanan, can you just give me
Just a minute, sir.
share my screen? Yeah.
Yes, sir. Yes, sir. Now you can share your screen, sir.
Yeah. Everyone can see my screen?
Yes, sir. It is visible, sir.
Okay. Okay, so good morning, everyone. I think it's a fresh day and a fresh morning, Monday for everyone. And everyone is safe and happy with their life. So before I start my presentation, I would like to thank Dr. Alagar Swami, Professor and Principal of MNR College of Pharmacy, for giving this opportunity. I will also like to thank Shri Ravi Verma, who is the Vice Chairman of MNR Educational Trust, and also the MNR Group of Trust for giving me this chance to speak with a wide range of audience belonging to several dimensions of career, and also the organizing committee for their valuable support. So welcome everyone. I think I'll just make the topic very simple so that it's easy for also the young people who are listening to this as for a new time, listening to a new dimension of stem cell therapeutics and diagnostics, and also to the people who are working already in this field. So today's my talk is going to deal with the nanotechnology, which is a new dimensions for stem cell therapeutics and diagnostics. So the outline of my presentation would be a brief introduction on the clinical bioimaging techniques, which is right now on the field. And what is the highly new, what we developed is a novel molecule, or we can say a novel nanomaterial, which most of them also heard as a quantum dots, which we say as a quantum, which is made up of a copper indium sulfide nanocrystals. And these nanocrystals has been developed for the first time in before, where we developed these nanocrystals for within a few minutes of process, and how we are going to use these fluorescent nanocrystals for labeling the stem cells. So stem cells, these stem cells are derived from the umbilical cord, which is being discarded as a medical waste after the birth of a child. And how we are going to use these label nanocrystals for non-invasively tracking the migration, and how we are going to see if they are able to regenerate the liver in living mice, in a live mice. And also I would like to extend my talk a little bit on how these MSCs, the label stem cells, could be also be used for diagnostics of early cancer in a tumor mice model. And finally, I'll go with the conclusion and what's my current research on. So before doing all this stuff, first we should know what is our vision and planning. So we should know what is our goal, what we really want to check. What is the requirement, how to start with that? Do we have all the materials and methods with that? And the next is how to develop it. We have all the accessories, but how to make and create the assays, which is really valid for our goals. So we should everyone know what to start, how to start, when and where, everything on goals, requirement on our development. And next thing is to have a stepwise analysis, and to not just, we don't really jump from just scratch and directly go to proving the concept. We need to have a stepwise analysis, and also there should be a multiple check on what we have to do. So most of us, we really do with the triplicates or duplicates of analysis, whatever we do. The next is the correlation in the findings. So whatever we have got the results or findings, we need to have a correlated findings. It should not be like certain assays are giving this kind of results and certain results gives you some other stuff. So we should have a correlation in our findings, so that are we right to the goals, and whatever we are developing, whether it's a nanotechnology based material or a drug or anything, it should not have any side effect, but it should have the right purpose of what it has been designed for. And finally, we should have these set of molecules to really show in a live animal model, so that becomes a proof of concept, so that they can be translatable into the clinical research. Okay, so this is the major a few common and general requirement where any young scientist should really think on how to and when to start with all this stuff. So let me start with an introduction. So we all are aware of the fact that there are various clinical biomedical imaging techniques which is available in the clinics in our hospitals. So most of the common one which everyone would have heard is the computer tomography, magnetic resonance magnetic resonance imaging, and PET, like, and SPECT, which is already being used for a lot of detection of cancer. But here, so in general, there are 4 biomedical imaging techniques. One is the optical bioimaging, and which is of high advantage because it has an highest sensitivity, and we can go for multicolor imaging, and it is a non-invasive technique. So you don't need to really cut off anything of the tissues in order to do imaging. Next is the computer tomography, where we make use of X-rays for imaging purpose. So the advantage of this is it has a high spatial resolution, and it can have a higher speed. And the third one is the magnetic resonance imaging, where we use of the radio waves. However, the advantages are they are harmless. They don't use any ionizing radiations, and it has a high spatial resolution. And the last one is the PECT or SPECT, which mainly use of the gamma rays. And we all know that gamma rays is an ionizing radiation, but it has certain advantages that it is highly sensitive, and there is no tissue penetration limit. You can go with any tissue depth for imaging certain things. So, and this imaging technology has been already clinically translated. So if you could see the examples below all of them, you can see some of the molecules which has been already designed in a nanomaterial. For example, MOS2 is molybdenum sulfide, and a lot of material which has been already designed in order to use it for particular bioimaging applications. If you go with if we go with if you see the sensitivity of the molecular sensitivity of each of the imaging techniques or the spatial resolution, we can see that the optical imaging falls in a very narrow path, where it can detect a nanomolar concentration of anything, and it can go up to 10 micrometer resolution. So that is the major advantage, where we can really explore to detect a smallest changes in the molecule within our body. Anything which is just emerging out, any changes in the biological system can be detected using the optical imaging techniques. And we can definitely see that with the increasing trend of the medical imaging techniques, we can see that with the 2024, we can see that there is enhancement in the preclinical imaging market over the years, and there is a huge requirement for diagnosing. First, we need to diagnose any diseases or any abnormalities in order to go for the further treatment. So that is the first case where we are seeing is the diagnostic. So in simple, you can see that intravital biomedical imaging is very important for tracking even the smallest biomolecule or any biological process which is happening in the living system. And among all the 4 imaging modality which you see up at the top, you can see that the fluorescent imaging is make use of a very low excitation energy, and it is generally from visible to near infrared spectrum, and you can image or visualize a multi-spatial scale that includes cells, tissue, or animals, and with a high detection sensitivity. So that is the advantages why I have chosen for my stuff is the optical imaging over the other imaging modality like CT or MRI, which are really into the clinic now. So if we can see the optical biomedical imaging, which we mainly focused on, it has a wide range of advantages. Like we can use it for live cell imaging, it has a higher resolution. It can be used for multidimensionality, and it is also versatile. Among this, if you all these 4 are unique for the optical imaging, and when compared to the magnetic resonance or CT. So if we can see, this is a Jablonski diagram. If people are aware of the fluorescence mechanism, how it takes place when a molecule is being excited, it gets from a ground state to the excited state. And when these molecules in the excited states come back to the ground state, they emit fluorescence. So that is what we are imaging in a detector. So if we can see that these optical biomedical imaging has a wide advantage in order to visualize even the dynamic biological process, not a static one, even the dynamic biological process, which is related to any physiology or any disease which is happening inside the system. So there is a huge requirement of these imaging system, basically the optical imaging. Uh, so there is an increased attraction of near infrared based optical bioimaging. I will let you all know about why we are more focused on the near infrared imaging, not the visible or the UV range side of the optic side. So why the near infrared bioimaging system is of more concern for the suitable, and it is affordable. And we need to have a better detection system so that even we can detect our smallest changes. And we should also have a strong probe which can really give you a strong signals for detection of the dynamic changes. And very important thing is we should have these systems, either a detection system or a probe which is out of any harmful radiation such as gamma rays, which we have seen in the previous slide that it is being used in SPECT, in PET or in SPECT, and X-rays which is being used in the CT, and ultraviolet radiations also. So on the other hand, stem cells based theranostic bioimaging has been growing with a very fast pace from the year 1965, where the first bone marrow stem cells transplant was discovered, and in 1985, which was a big year where the first blood stem cells was transplanted for the Fanconi anemia patient and for the clinical trials. And now we can see that in year of 2020 and 21, there are more than 80 plus diseases, and there are more than 200 to 300 clinical trials, which has been already been going across the world for the treatment of several diseases. And what all these diseases are, because these stem cells have a potential for the treatment of a lot of things, like if you can say baldness, blindness, deafness, myocardial infarction, muscular dystrophy, diabetes, which is most common, and the cancer, even osteoarthritis, and definitely wound healing, and very rarely and very importantly, the Alzheimer's disease and even the Parkinson's disease, which is where these stem cells have shown a very potential application for the treatment of all these diseases. And the reason behind this is they can home at the site of the inflammation, and they can regulate immune responses. They regulate the blood vessels growth, and they reduce the scarring and prevent premature cell death, repair and regenerate the tissues. So although these stem cells have shown a promising preclinical result, but even though, there are only few applications which has already definitely reached into clinic due to the safety and efficacy concerns. As I've told you that there are more than 200 in clinical trials, but there is like only for few of the treatment, like I can say that osteoarthritis, stem cell therapy is being started in the Apollo hospitals, where we use stem cell therapy for the treatment of arthritis. So there are few of the cases where stem cells have crossed the clinical translation, but it's not that all the subject of diseases has been really approved for the stem cell therapy. So the major problem with the stem cell therapy is like when they inject the stem cells in a doctor inject stem cells into a patient in order for the treatment of the purpose. The stem cells, any doctor doesn't know how the stem cells go inside the body. They inject into the blood or intravenous. They don't know how any person can personally cannot find it out how these stem cells are going inside the body. So here is the material and nanotechnology which has come into the play, where it has been used for lot of domain stems in the stem cell research. For example, polymer scaffold, it has been used as a cargo or as a base material for the transferring of the stem cells, and other is liposomes or micelles, and inorganic nanocrystals, which I am going to focus on. And there is also collagen nanofibers, graphene, and scaffolds, and peptides and hydrogels. So these are the lot of nanomaterials which has been being explored in the stem cell research. So here now I would like to just spend 2 minutes on that. So what is actually stem cell labeling is? So there are two types of labeling. There is a direct labeling and there is an indirect labeling. So what is direct labeling? Direct labeling is mainly used of an external synthetic material or nanomaterial which is synthesized in the lab, and then we treat it with stem cells in order to label them. So that is the direct labeling. So this all are the inorganic or organic materials. For example, this is IONP is ion oxide nanoparticles or quantum dots, or all these things are the SPECT and SPECT based the contrast agents, what we also say in the clinical terms. So these are directly incubated with the stem cells for direct labeling purpose. And what is indirect labeling is by labeling the genome or the gene of the stem cells. So we everyone would have heard about the GFP, green fluorescent protein, or we also, so what we do is any cell, all the cells, we always have their transcription and translation process happening inside it. So we used to label the GFP with the most translated or transcribed and translated protein, so that whenever the protein is being expressed, it expressed with the GFP, and thus the cell is labeled with the green fluorescent protein, so we can observe the green fluorescence in the cells. So that is the indirect labeling, like the labeling the cells in the level of genome. So once the labeling is done, these stem cells are glowing now. So what we do is we culture them. Okay, we culture them in the 2D culture plate, and then we characterize them, because these stem cells should have their ability to proliferate and differentiate. That is their inherent or intrinsic properties. So we should make sure that their properties of doing things are not changed. So that we do characterization. And once we assure that even after the direct labeling or indirect labeling, their property is well safe, we go for the transplantation or we injection into the living model, and we find and then finally we go with the imaging of these stem cells, so that we can track whether these stem cells have gone to the brain, or it gets gone to the heart, or it's gone to the liver. So the degree of location of these stem cells which is being injected in the blood can be traced out using the different imaging modalities. So that is what the whole concept is. So before we started doing all this stuff, this was just in the books or in the theory. So there was a major challenge in order to how to track these stem cells inside after their engraftment, and also how to check their proliferation and differentiation inside the living system in order during their regeneration or diagnostics or therapeutic applications. So then we also, it is important to develop an high resolution imaging system. As I said in the first slide, the optical imaging gives you the highest resolution in terms of other than the other imaging modalities. And we also need to develop a nanoprobe, because the major problem with the optical imaging, why it has not come into the clinic, is because of the limited nanoprobes or the contrast agents for the optical imaging, in order to better understand the therapeutic mechanisms in the regenerative medicine. So if you can see, and I think many people would be aware of the fact that we have a lot of conventional fluorophores, which has been already into the different profile of biotechnology, biochemistry, and microbiology, where we have a lot of dyes which we use for labeling and staining cells. So, but there is a few problems with the existing dyes. Is like they make use, few of them make use of high energy excitation, that is UV, and as we know that UV also lead to a dimeric formation in the genes. And other few of the main problems is the photo bleaching effect, where when we irradiate the dye for a longer period of time, they bleach up, and there's also an overlapping excitation and emission spectra, as you can see over here. And most of the dyes also show an autofluorescence, which leads to a lot of background noise during the imaging system. And third is they have a limited penetration depth. Most of them, there are few which is gone with the more penetration depth, but in general they are having a limited penetration depth for the deep tissue visualization. And finally, the very important one is the phototoxicity, because since when all the dyes is being exposed with the UV light for the detection, they undergo some structural changes which directly or indirectly affect the biological process inside the cells. So that is few of the known and well-documented fact about the fluorophore. But when we go into the nanotechnology, initially, like in 2010, like there were bundles of publications which was pushed up for the development of new nanoprobes. And all these nanoprobes or contrast agents for optical imaging mainly consisted of cadmium, lead, mercury, selenium and arsenide. So I mean, everyone know that these are the heavy metals, and however, these elements have shown that high photostability, which is high broad emissions tunability and high extinction coefficient. And they have also shown been used and been proposed for different applications like metal ion sensing, drug delivery, molecular detection, and bioimaging applications. But the major problem is, as we can see, these all heavy metals have a potential toxicity. Either it can be acute or chronic in the living systems, and also once they are disposed into the environment, it is a big medical concern. So there was an, when I started with doing my PhD, there was a huge demand to have an alternative to all this heavy metal based quantum dots, what they have been told as, and then in order for the optical imaging. So we try to design a material or nanotechnological material which is devoid of cadmium and lead. So here we propose, I mean, I'm going to come with the material of it. So all of these heavy metal based materials, metals or alloys has been proposed, and though they have different advantages and drawbacks in terms of their tunability and purity of emissions or quantum yield, fluorescence, quantum yield, lifetime, and photostability. So here we proposed with, we came up with a new molecule which was really safe, and it has all the advantages of the for the optical imaging system, and it was not at all toxic to the human being. So it is copper indium sulfide nanocrystals, and it is physics of it is it is a direct semiconductor nano material, and it has the extinction coefficient of is in the visible spectral, so that you can, if you want to tune, if you want to have a blue color quantum dots of fluorescent material, you can have blue, you want green, you can have red, anything. So you can just tune with that, whatever color of fluorescence you need, you can just tune this material in order to get a different fluorescence stuff. And very importantly, as I told, the dyes have an overlapping spectra. These material have a larger Stokes shift. That means it has an the emission and excitation and emission spectra is widely away from each other, so that there is no overlap, so there is no autofluorescence, and there is a lot of more advantages. So the advantages of this material was that you can even tune the size of it. That can give you different fluorescence. You can have a different composition tuning. And there was also the third is the surface tuning, where you can always play with the surface of the copper indium sulfide with the zinc sulfide, so that you can really play with the colors of it and also enhance the stability of the molecule. So all these things can reduce the amount of surface trap, because surface traps are the ones that reduces the fluorescence of a particular nanomaterial. And also they also reduces the non-radiative recombination. So now, very importantly for one and everyone here is why we choose the near infrared over the visible and the UV region. Because in the near infrared stuff in the region, if you can see this graph, within from 200 to 600, we can see that there is a lot of absorption of molecules, absorption of the light, which is being taking place before 600. So we choose a domain which is around say 600 to 1400 or 600 to 1000, where you have a you have a reduced fluorescence here. So we choose, I mean, so that means if you have a reduced absorption of your fluorescence inside the tissue, you get the enhanced signals in the detection system over the living system. So that means you will get a better image when you have the lower absorption in the tissue, and there is more of reflecting. So we choose to have the first biological window, which is around like 650 nanometers to 950 nanometers, which we say the near infrared first frame. And because it minimizes the light absorption and diffusion by the molecule, which is relevant to the biological systems, like even it can be water, hemoglobin, skin, or fat. So that is the reason why we are more focused on the near infrared over visible and UV. So how we went with this is we designed these molecules using a simple one-pot method, which is which was just 6 minutes of preparation in the domestic microwave oven, which we all have also in the home. So the advantage of this is this was a novelty actually. The reason is all this material has been proposed already long years back for solar cell application, but however, this doesn't show any fluorescence. And the synthesis of this material generally takes more than 3 hours. And the novelty over here was we designed this material in just 6 minutes rather than 3 hours. So that was a very quick and usable stuff for the clinical stuff. So here is the simple process as schematically represented. We use the precursors in the solvent, and when we heat it in the microwave in a particular system, we get a red fluorescent solution, and this red fluorescent solution is called 1-DDT functionalized. This is an organic solvent dissolved nanocrystals. So as we all know, when we go with the biological system for clinical translution, we need a water-soluble contrast agent. So we do a ligand exchange process where we developed a water-soluble red fluorescent nanoprobes or contrast agent. So that was one of the novelty. So not going with a lot of synthesis process. So this was optimized with different reaction times and everything in order to develop this. So as I said in the first slide, we need to go step by step. When we design a molecule, we need to characterize them in each and every step in order to find out whether the molecule which we have designed, we really need it or really in concurrence with what is expected to do in terms of function. So we also characterize this contrast agent using XRD and XPS analysis to find out whether these molecules are really formed in the way they have to be or not, or is there any side products which has been formed along with that. So we confirm with that. In order to also confirm the ligand exchange process, I said in the synthesis part that whether these molecules are, which is in the organic phase, is being transformed into the water soluble phase for the biological application. So we confirm using the FTIR and the NMR analysis. So for the many people who have not seen how an atom looks like in real, so I just want to show that this is the red fluorescence which is coming from the contrast agents from the nanoprobe which we designed it, and this is how they look like in an high resolution transmission electron microscope. So as we go over the magnification of it, we can really see this all the single dots, white dots are nothing but the atoms. So this was very much interesting to know that these we synthesize a nanocrystalline contrast agent which has very high fluorescence, and this is the reason where and the size of these nanocrystals were just 6 nanometer. Okay. Then we also, since we are speaking about fluorescence, we need to find out how much the fluorescence quantum yield is. So we found out that for the material, the nanoprobe has a high quantum yield of 76%. But the very interesting part over is the time resolved photoluminescence. So we use a lot of dyes in our own labs and experiments. We can see that all these dyes have a very short lifetime of 5.46 nanoseconds. So what this lifetime means? So lifetime means if a molecule, if a fluorescent dye is being injected with 1 photon, it should emit 1 photon. But the time it takes to emit 1 photon determine the lifetime. So for a dye, it takes only 5.46 nanoseconds and it just emit very fast. So what happens is you have to give a lot of photons in order to get subsequent images. But what happens with the nanomaterials which we develop is it has a lifetime of 0.6 microseconds. So it's more than 600 to 6,000 times. So what happens is if you give 1 photon, you can image it for a very long period of time. You don't need to give more and more energy every time in order to do the imaging aspect. So that was the novelty of this molecule. So rest now brought below is the physics of it. Let's not go with that, but that really tells that there is a better fluorescence of these nanomaterials, and it has a higher lifetime so that you can use it for a long period of time. So yeah, so for the people who are really waiting for the biological stuff, what is really it's going to be translated in the biology. So till before last slides, we just seen how we are developing contrast agents, how we synthesize a contrast agent, how we characterize this contrast agent, and how we make sure that, yeah, this is going to really work in a biological system. So what we did is first we isolated the stem cells. So we isolated the stem cells, as I told you, from the umbilical cord, which is generally medical waste which is thrown out once the baby is out of. So this is the stem cells which really look, they look like in the phase contrast microscope. So what we did is first, in order to go before using them for labeling purpose, we need to know whether are they toxic to the stem cells. So we checked with the cell viability, and also the we also checked the ROS, that is the reactive oxygen species, which has been produced in these stem cells. And so we found out that even after an incubation of more than 372 hours, there is no death of it, or there is no enhanced more reactive oxygen species being developed in the stem cells. So as everyone know that these ROS, or reactive oxygen species, are very important to be characterized, because these ROS, if the concentration of RO is high, the cell undergoes apoptosis and they go as a programmed cell death. So the stem cells will die if the ROS level goes up. So that's the reason we should also check the ROS concentration of it. And the fourth one is the flow cytometry analysis of. So okay, once we have found out that yeah, stem cell is safe, it's not being toxic to the stem cells, the contrast nanoprobe which we have developed. Now we have been using for the imaging, for labeling aspect. So we, in order to know how much is the percentage of labeling, we checked using the flow cytometry analysis. So here we can see that there is a 98% of labeling. That means out of 100 cells, there is 98 cells which are being labeled using this nanoprobe. So that was the very high amount of cells which is being reported ever before. So yeah, so here we got a quantification of it, how much is that. But really seeing into the eyes, here we can see that we have 4 set of image. The first image is DAPI staining. DAPI staining is used for staining the nuclei. F is the phalloidine actin staining, which is used to stain the cytoskeleton of a stem cells. And the G is the red fluorescence, which is coming actually from our nanoprobe, which we have synthesized in the previous slides. And H is the overlay image of all the 3 before. So we can really see that these nanoprobes are really inside the cells, and they are not just outside or as an background fluorescence. So that is how these images were captured in a confocal microscopy, and we could really confirm that yeah, they are having a high labeling efficiency, and they are having no toxicity, and they are also able to label the stem cells, right? So now, once these stem cells are labeled, now we should know if, as we know that every stem cells, in order to once they are going inside the microenvironment of certain injured tissue or any inflammatory site, they should interact with another cells. So each of the stem cells have a set of surface markers around them, which will interact with the neighboring cells in order to communicate either it's a proliferation or any differentiation or regeneration of anything. So we also need to check whether these surface markers are not affected despite the labeling of the MSCs of the stem cell. So we also check the expression markers of these surface markers using again a flow cytometry, and we found out that the labeling, there is no significant change in the expression of these surface markers even after the labeling of the stem cells. The reason is we need to check in the cell level everything, because before we go into the living mice, where we have to make assured that they are not toxic and their expression markers are safe, everything. Now, once we have assured that these stem cells are labeling fine and they are proliferating fine, now, as everyone knows that stem cells have an ability to differentiate into a specific tissue. Either if they go to the kidney, they have to differentiate into kidney. If they go into bone, they have to differentiate into bone cell. If they go into fat, they have to differentiate into fat cells and so on. So we also check the differentiation ability of the stem cells. So after the labeling of the stem cells, we should make sure that they are having all the differentiation process as they do in a normal controlled cases. So we found out that yeah, their adipogenic differentiation, osteogenic differentiation, and chondrogenic differentiation were well safe using the RT-PCR analysis. So that was quantification, and now the imaging aspects, we also confirm the differentiation of these stem cells using the histology staining, which is conventional staining which we use. And for adipocytes differentiation, once the stem cells differentiate into adipocytes, we can see here the control one, and here this one is the nanocrystal label stem cells. We can see the expression of the red part is the adipocytes, which is got differentiated. This we do with the picrocyrus red staining. We can also see the adipogenic differentiation using these lipid droplets. So adipocytes show a lot of lipid droplets. So these circles are nothing but the lipid droplets. Now, if these stem cells are differentiated into osteocytes, that is bone cells, we checked using the alazine red staining, and we also using the alazine red staining, which really chelate with the calcium 2+. And in order to see if there is a chondrogenic differentiation of these stem cells, we checked using the toluidine blue staining. So these are the conventional staining process, which is well known and well documented in order to see if the normal stem cells are same as the labeled stem cells are also. So that is how, so we need to have a step-by-step characterization so that we make sure that, because once they are injected or transplanted into the living system, it doesn't misbehave or change its functionality, or they fail to do the function for which they have been transplanted for. So now, once we have labeled it, as I told you all in the previous slide, that they are able to have a labeling efficiency of 98%. Okay, so that 98% was once they are labeled. But there was a question like, how long does this labeling stays for? So we checked again using the flow cytometry analysis, where we found out like the labeling efficiency was like once they are labeled, they stay for more than 7 days. So that means if you are labeling a stem cells, it will stay for 7 days and it will do its function on its own. Now, then we did some of the other experiments that we call as transfer experiments. As we all know that when a cell is being given with the external moiety, a drug or a dye is a small molecule, but when you go with a nanomaterial being labeling for the stem cells, it is like a carrier. So when these stem cells also should have a migratory behavior. So once the stem cells goes into the microenvironment, they should also be able to move across the microenvironment. They should be able to migrate from one place to another in order to do the regeneration process or healing process, everything. So we also check whether they are having the migratory behavior is not lost when we are labeling that. So that we do using the transfer experiment. There was one important question about if once we are labeling these stem cells, are they not coming out of these stem cells, or like what the process we say as exocytosis? Is there any exocytosis of these nanoparticles since they are foreign to the cells? Are they pumped out of the cells? So we again do confirm that using the transfer experiment, where we can see that here, which is the upper region, we can see there's a lot of red fluorescence which in the middle, which is coming from the stem cells. But here, there is no fluorescence detected here. That means there is no leakage of any nanoparticles from the stem cells. So that we confirm using the transfer experiment. So this is all the basics of the in vitro level studies, where you do a lot of cross check with the nanoprobes which you have developed with the stem cells alone. But now when we inject these stem cells for the purpose of the regeneration, that is to regenerate the liver, we go with the mice model. So here we can see that going over here to the video, here is the injection point. So once we inject this label stem cells, which has a red fluorescence, near infrared fluorescence, it goes to a point over here first. So this point first here is the lungs. So every stem cells have an ability to first go to the lungs. When they are going in the blood, they first cross the lungs, and there is a transient pulmonary trapping, what we say as a mechanism, where there is these stem cells, few of the stem cells are trapped in the capillaries of the lungs. And that's why there is the first sign of the fluorescence which is observed in the lungs. But since these mice is a liver injured mice, there is the migration of these stem cells from the lungs to the liver. So this part is the liver. And we can see that over the period of time, there is enhancement in the fluorescence because it is the regeneration process which is taking place. So what we seen over here is in the different time lapse image. Here we can see that first we see in the lungs, and over the period of time we can see the distribution of these liver stem cells going into the liver. And after the second day and third day, we could see that there is no fluorescence, and there is the complete emission of these the fluorescent nanocrystals out of the body at the end of the third day. But are we sure that these nanocrystals have these stem cells which was transplanted has really regenerated the injured liver in these mice? So what we did, before going to the characterization of that, we found out whether these fluorescence is really coming from the liver or is it coming from the different part of the body. So we do an ex vivo imaging where we take off the every organs, and we found out that this fluorescence is really coming from the liver and not from the other organs. And there is a little fluorescence in the lungs that, as I said, it is from the pulmonary transient trapping. Then, in order to confirm the distribution of these nanocrystals or these nanoprobes in the liver, we do a confocal imaging where we take the whole liver, we cut into different section, thick section, and here we use a 35 micrometer thick section of the liver, and we do a confocal Z-stacking imaging where we find out that there is a distribution of this is red fluorescence within the tissue of it. So this red fluorescence is nothing but coming from the nanoprobe which we have synthesized before. Okay. So now, in order to confirm, yes, we have seen that the liver is being glowing with the nanocrystals, and it is having the stem cells. But whether this liver has really regenerated or not, we are doing it using the histopathology analysis. So where we can see that, we'll just focus on here, the liver. We can see that this is a control liver, which is very perfect with the architecture. But once it is being damaged, it looks it's like a lot of necrosis which is being visible in the injured liver tissue. But once when we transplant the stem cells, we can see that there is a regeneration of liver which is taking place. So here we can see that it's either way. We have achieved technology where we can really track the stem cells moving in the different part of the inside after the transplantation. And also second thing is we can also confirm that there's a regeneration of the liver which is taking place in the injured mice. So in order to confirm whether not in the tissue alone, also in the blood, because we have a lot of different function tests which we say is a liver function test, kidney function test, in order to know whether there is any injury or any change in the physiology of the human body. So this is the liver function test where we can see that this green is the control one, and the red is the injured mice where we can see that when the liver of the mice is injured, the level of AST, ALT, and ALP goes high. But once we transplant these stem cells, we can see that there is the reduction in the levels of AST, ALT, and ALP. So that shows that there is a regeneration process which is taking place, also confirmed in the blood, not alone in the tissue. So once we confirm that, so we have seen that these contrast agents which we have developed we developed in a very short period of time. It is safe with the labeling with the stem cells, and also we have seen that it can be tracked once they are transplanted inside a living model. And we also seen the fourth thing is that they can really regenerate a particular injured tissue, and using the histology analysis, and also we confirm the cross-check with the blood biochemical analysis, hematology analysis. So now we extended again this nano contrast agent, whether we can again tune this molecule in order to can be used for the different imaging modalities. So what we did is we incorporate some of the moieties for the computer tomography imaging and also the magnetic resonance imaging. So what we get out of all this thing is a modality technique where particular nanoprobe can be used for all the three imaging modalities. So we use this multimodal imaging probe or contrast agent for the diagnostic of early cancer. So what we did is again coming back directly to the in vivo model studies, where we can see that once we use this contrast agent, this is a tumor site. So when we inject these multimodal contrast agent, they are accumulated in the tumor site. The reason is the tumor has enhanced permeability and retention effect. where these molecules go and get trapped because there is a lot of vascularization which is inside the tumor cells, and these nanoprobes have a huge access to the tumor when compared to the rest of the tissues. So we can see a bright fluorescence which we could detect in the tumor site. Also, we confirmed the fluorescence, is this really coming from a tumor, by ex vivo imaging, where we take off all the organs and we find out that yes, this fluorescence is really coming from a tumor and not from any other organs. The liver shows light fluorescence because it is most often that liver is it's a first pass metabolism where most of the thing crosses the liver. So there is a light fluorescence which has been observed in the liver. But the majority of fluorescence here, it's only red, but here the majority of fluorescence which shows that it is blue, which means that there is a huge concentration of these nanoprobes or contrast agent being accumulated in the tumor, so that it can be used for the early diagnostics or detection of the cancer. In order to find out the distribution, again, as the previous slide, we did the confocal imaging of the thick section of the tumor, which is 30 micrometer thick section of the tumor, and we could see the distribution of these nanocrystals in the tumor site. Further, we also confirmed whether these nanoprobe, which we designed for the multimodal imaging, can be used for computer tomography and also magnetic resonance. Yes, we were successful in showing that this is the tumor site. This is actually an axial view where we can see that there is a tumor in this spot, and we could also detect the red fluorescence which is coming from the contrast agent, the nanoprobe which we transplanted into the mice using the computer tomography. So even the coronal view also showed up the signals of the tumor. We also confirmed this the tumor, the distribution of tumor or the presence of tumor also using the magnetic resonance imaging. So generally magnetic resonance imaging have two images, T1 and T2 images, in order to differentiate and contrast between with the microenvironment of the tissue. So we can see T1 images gives you a brighter images and T2 images give you a grayscale images. So T1 images really showed a bright signals in the tumor spot, and there was a gray signal which we detected in T2 images. So here we can really confirm that, yeah, so whatever nanoprobes or nanocrystals we have designed, which was a very short period of time, is being used for both tracking them inside a living system for regeneration, and also can be used for extended for the diagnostic or detection of cancer. So we confirmed again using the histopathology analysis, and also in order to further show whether these stem cells are in the tumor, we also carried out the immunofluorescence studies and the subsequent parallel studies in order to really cross-check and confirm that these fluorescence or these detection signals are really coming from the site of our target and not from the background or from the microenvironment. So we did also the 3D surface plot and also ICP-MS analysis. Also, we confirmed that even after the transplantation of these stem cells, the hematology analysis of the mice is not being altered using the blood biochemical analysis. And we see that there's an optimal concentration of all the molecules, which is in well agreement with the control group. And but there is few variations that is mainly due to the tumor associated metabolic factors, because when there is a tumor presence of tumor, there is a changes in certain molecules. For example, there's an enhanced concentration of GGT, which is due to high GSH metabolism, which is happening in the melanoma cancer. And there is also enhancement in the C-reactive protein, which is due to the progression of the tumor. And so that is the case where we can see the little bit of the enhancement which is happening. So yeah, so this is a huge part of stuff which is being just briefed in the few slides. So what we have seen over the last few slides is we developed a simplistic labeling protocol, which could be a way for so many researchers to effectively track the rare MSCs or rare stem cells that are really challenging to transfect or express fluorescent proteins or non-fluorescent material to monitor their fate inside the living system. So the future work is more focused on translation of these multifunctional nanoprobes or the contrast agent for the multi-clinical imaging modality for the diagnostics and therapeutics of several real-time applications. Also, these nanoprobes or contrast agents can give you a deeper understanding on several brain diseases like Alzheimer's and Parkinson's diseases to track brain-specific molecules like such as amyloid beta and dopamine, etc. And if we could optimize the preclinical imaging configuration, it can increase the applicability and enhance the signal to noise ratio, because that's too important. And it can allow the state-of-art studies for widespread applicable use. And very importantly, currently we are also exploring the complex heterogeneity within and among the microenvironment, like for example, the regenerative tissues, injured tissues, or apoptotic tissues, or necrotic tissues. And we are also focusing on a design of precision nanomedicine towards the personalized regenerative treatment. And in more particular, we are more into the development of a viable and non-invasive long-term therapeutic technology for diabetes and multi-drug metabolic induced kidney injury. So last but not least, I would like to thank all my PhD institution and my collaborations in all the institution for all the sets of work which I have personally been to all the institute to carry out all this work. Last but not least, a glimpse of the Stanford University here. So the first is the Stanford University, and this is the School of Medicine. We have a lot of work which has been going on for a lot of diseases, including stem cells. And this is the Department of Chemistry, where people are working with designing and developing a lot of molecules for a lot of purpose, even organic fluorophore to the thernostic molecules. Thank you all for listening. And if anyone have any doubts, and they are always welcome to ask me either here or in my mail ID. I mean, any kind of guidance. Thank you. Thank you all.
Thank you, sir. Thank you, Dr. Sasank Sethi, for the wonderful presentations on nanotechnology, a new dimension for stem cell therapeutics and diagnostics. So from your presentations, we came to know why you are so much comment on over that nanotechnology. So it is definitely an eye-opening session for all our budding pharmacists. And those who are having any queries, you can raise your hand. I will unmute it, and you can directly ask. You can directly interact with Dr. Sasank Sethi. Please raise your hand. I will unmute it. You can directly interact with that speaker.
I think there are a few of the questions in the chat.
Yes.
Meantime, if anyone has, but I'll just go through with that. So there is one question like, can you explain how it is helpful in Parkinson's and Alzheimer's disease? As per my knowledge, Parkinson's is due to drop in dopamine levels, and yet there is no known diagnosis being found to it out. Please help me out. So here what we are doing is designing a fluorescent molecule so that we can track it, we can tag it, we can label it. So if you can tag and label your dopamine, or you can genetically engineer the excreted or translated dopamine with a fluorescent molecule, you can really track its functionality so that you can really know how these Alzheimer's or Parkinson's disease is really the molecular mechanism behind how this whole process takes place. So once you know the whole process, how this mechanism takes place, you can really design design your molecules, or you can design any treatment, diagnostics, or therapeutic technology with that. So I hope this answer is fine, but if not, you can again mail me back. Next question is here. Why is stem cell following the route, like from lungs, then liver, spleen, and so on? Yeah, seriously, excellent. Okay, so this is the root of the stem cells is an intrinsic property. What we say once, so these stem cells are being injected into the intravenous of the living system. So once they go into the blood, so as we all know that the blood first, when it's going to the lungs, there is an exchange of oxygen and carbon dioxide. And everyone knows that lungs has more of capillaries, wide volume when compared to the other organs. So what happens is when there is a wide vacuum volume, these stem cells, when they are moving through the lungs, they get trapped in that wide volume sometimes. So that is a process called transient pulmonary trapping. That is the intrinsic property of the stem cells. So whenever we inject stem cells, it's one of the property, and it's also one of the concern, I should say, because if you are injecting, for example, 100 stem cells, few of the stem cells are trapped into your lungs, and only the limited number of stem cells really reach the target you are really in need for. So that is also one of the concern, but that is one of the intrinsic properties. So in order to avoid, yes, so every challenge has one solution. So in order to avoid that, there are a lot of few molecules which has been proposed in the few past years. Like one of them is heparin, which is being injected along with the stem cells in order to reduce the transient pulmonary trapping and enhance the targeted homing of these stem cells into the target site. So that is one of the, and there are many other molecules which has been also been tested in order to reduce the traps and then make the stem cells directly go to the purpose for which they have been transplanted. And next is why we use UV rays or gamma rays in CT scan and PT, even though we know that it's harmful to our body. Yeah, so that is these all PET or CT, everything has been designed for own being clinically used, and it is been designed. That is the principle of the system. It how the principle of the working of the system and how it works. So why we use and still why it is being approved to use is just because um, we are not being exposed, any patient has been not exposed for a longer period of time for a particular thing. For example, if anyone have done CT, it takes say few minutes of that, and also there is lot of constraint that you have not move yourself. You should have to be static. You should during the CT, it is a place where you have to hold your breath for a longer period of time, and then till the scan is complete, then you leave your breath out. So there is, and that is also applicable for MRI. But the point why these CT scans and these imaging modalities being approved for clinical purposes, like they give you a better image and give you the right the diagnostic resolution. So that is the reason they are really into the market or into the clinic. But unlike the optical imaging, where I am personally focusing on, is not into the clinic, but I am trying to make sure that to develop a better nanoprobe, a contrast agent, and also to have a better equipment so that it can give you the right resolution as the PT, CT, MRI, and every other imaging modality is giving. So we should always have a better contrast agent also, and also the better imaging system. So both need to be parallel. So that is the one thing which that's the reason why optical imaging is not right now clinically into the in the hospitals. Also, the next is, okay, I think there's a lot of questions from Mona. Okay, also, sir, if stem cell therapy is so beneficial, why isn't it handy to many people suffering? Yeah, so stem cell therapy is being it's very beneficial, and it is under a lot of clinical trials, and it has been approved, and there are a lot of therapy which has been really going globally. In India, it is taking time, but there are a lot of, to my personal knowledge and to my seeing very in person, is a lot of the private hospitals have really taken up stem cell therapy for the treatment of several diseases. As I mentioned, one of them which I've seen and personally is like the Apollo hospitals, which really work with the stem cell therapy for the arthritis and many more. And yeah, next question is, what is the harmful effect of nanotechnology? Yeah, so in simple terms, anything beyond the critical limit is toxic. Anything beyond the safe, below the threshold limit is always safe. Either it can be a drug, or it can be anything, any habits or any stuff. So anything beyond the critical stuff is always harmful. If you really work in the threshold limit, even if it's a nanotechnology or physics or chemistry, everything works fine. There's the same question from Pratibha. Okay, why stem cells first enter into lungs initially? Okay. Then next, how long will a stem cell therapy take to show its effect in the body and complete the therapy? So to the studies which we have done, the stem cell therapy, we work with the acute liver injury mice model, where there is a acute injury in the liver. So for us, it took 3 days for the liver to regenerate. As you know, the mice has a very small liver. So it took just 3 days for the regeneration of the liver, and we could see the reflection of the regeneration using the histology staining, and also we confirm using the hematology blood biochemical analysis. So, but different diseases model have different regeneration capacity, and they have a different reflection of their regeneration which has been taken place. The next is, does stem cell therapy in heart failure improves ventricular injection fraction? Yes, there is a lot of work. As I said in the previous slide, it's been a lot of work which has been going on the cardiac muscular dystrophy, and a lot of work which has been really going in the heart cardiac tissues. I want to know about the recent updates in stem cell therapy, possible uses, challenges. Yeah, so uh, there are a lot of work which has been done with the stem cell therapy for the diabetes, which is to improve diabetes. But right now, the major challenge with the diabetes for the stem cell therapy, or any other therapy, I should say, is the mechanism, the molecular mechanism behind every therapeutic action of the stem cells for particular diseases. So that is the main target, main research which is really going on to find out the molecular mechanism behind everything. What is the signaling pathways that are upregulated or downregulated during the process when we inject stem cells or we don't inject stem cells? What is in the injury case, and what is in the stem cell case, and what is after the regeneration has been happened? What is the molecular mechanism? And next is, the expression of markers are compatible with the in vivo environment. Yes, they are compatible with the environment. And the last one, there is several autoimmune diseases, right? So in what case, how, and what type of cells does stem cells mimic in order to benefit the patient? If it is gonna mimic the one which is autoimmune, the one is destructing, so could not be any problem rising in that case. Yeah, so there is again definitely a lot of immune responses which is being modulated by stem cells, because stem cells are known to do a lot of functions. As we know that stem cells are the primary cells, they can differentiate and they have a functionality to do a wide spectrum of function when they go inside the cell. So they really are a lot of field of work which is all really going in the autoimmune diseases also. Again, last, what is the difference between therapeutic cloning and reproductive cloning? So again, this is not the work area where I'm working on, but yeah, I think you can read up yourself. Thank you. Any other questions from the audience? You can feel free to ask, or you can even mail me or anything.
Thank you, sir. So if anybody have any other questions, you can raise your hands and I will unmute you. You can directly interact with Anna, sir. Otherwise, we will conclude that sessions.
There's one thing, why not use adult stem cell instead of using human embryonic stem cells in the research? The research is going in all the aspects. I will say that adult stem cells is being used, embryonic stem cells is used, and there is now, again, I think everyone are well aware, is induced pluripotent stem cells. And so there is different branch of stem cells which is being used for different applications, and all of them have different capacity of doing things.
Yes. Thank you, sir.
There's one more. Sir, can a dead cell, necrosed one, can be replaced with stem cells? Yes, that is the reason where we are really working on. When we do a necrosis, particular tissue is under necrosis, these stem cells can go to rescue these cells or replace with or differentiate into the particular cell type, or it can stimulate the proliferation of existing cells. So there are many things which can happen. It can differentiate, or it can help the neighboring cells to proliferate more in order to fill up the necrosis. Thank you.
Thank you, sir. Thank you so much, Dr. Shashank Chetty, sir, for enlightening us with the new dimensions of nanotechnology for stem cell therapies and diagnostics. So as a token of appreciation and as a token of love, we are presenting one virtual certificate and a virtual memento, sir. Please accept that one, sir. Mr. Ramanand, sir.
Thank you. Thank you. I should stop sharing, I guess.
Yes, sir. Yes.
Yeah. Thank you. Thank you. Thank you. Thank you, Dr. Professor
Thank
Alagan.
you, Dr. Sasan. Kindly accept this certificate as a token of our appreciation.
Thank you. Thank you.
And
Thank
also
you.
kindly accept this virtual memento.
Yeah, thank you.
And as you promised, you will be visiting our institution soon when you are visiting India.
Yeah. Yeah, definitely. Definitely. Yeah.
That time we'll give you the laminated this memento. Kindly save this
Thank you.
virtual memento. And thanks a lot for sparing
Yeah.
your valuable time with us. So
Thank you.
definitely it will be a long way to go, and we'll have a collaborative research in the coming days, and we'll do something for the community for the suffering
Yeah.
peoples, and we'll have
Yeah,
soon
exactly.
have further interaction regarding our collaborations.
Yeah. Sure, sure. Yeah.
Thank you very much.
Thank
You
you
can
all.
stay for some more lectures,
Yeah.
which is coming up in 5 minutes,
Yeah.
and we can
Okay.
view and you can witness all those lectures also.
Yeah, sure. Just FYI for all the participants, if you have any queries regarding the research or even a guidance of what to do next or any kind of stuff, you can always mail me. So I'll be always happy to speak with the young budding scientist, or an opportunity to share what's available in the US or any other countries. Thank you.
Thank you very much, madam. So, Dr.
Thank you.
Saravanan, we can go to the next session. Madam Sai Jyotsna, madam, we can go to the next session. So, let us see whether Dr. Carlos joined or not. We'll see.
Sir is joined, sir. Just now, sir is joined.
Okay. Madam, you can proceed to the next session.
Okay, sir. Okay, sir.
Madam, just a minute, I will encourage. Dr. Carlos. Dr. Carlos, please unmute your mic, sir. Dr. Carlos, please unmute your mic. Dr. Carlos, please unmute your mic, sir. Dr. Carlos, please unmute your mic.
Yes. Hello. Hi.
Yes, sir. It is audible, sir. Welcome, sir.
Yeah. Yes. I have succeeded. Yeah. Hello. Hi. How are you?
Fine, sir. Let me start our program, sir.
Yeah. I succeed to enter.
Yes. Okay, sir. Madam Sai
Okay,
Jyotsna,
sir.
now we can start that session, madam.
Okay, sir. Now I invite Dr. Sandhya, Assistant Professor, Department of Pharmacy Practice, to introduce the next speaker, Dr. Carlos.
Thank you, ma'am. I take pleasure to invite Dr. Carlos Hoyo Vadillo, sir. Next slide, please. Dr. Carlos Hoyo Vadillo, sir, who is specialized under Department of Pharmacology and Toxicology, Silvestre, Mexico in 1989, has done a great publications and was one of the a great person who has done postdoctoral stages. He has done various postdoctoral stages at University of Leiden, the Netherlands, and GSF Center for Research in Environment and Health, Germany, University of Connecticut, USA, University of Navarra, Spain. He has also done his master's in physiological sciences and bachelorate in biology, Mexico.
Yes. Thank you.
He has produced 43 papers in recognized journals 4 chapters in books, 13 reports of investigation, 4 computer programs, and 1 indulging paper. He has participated in 5 courses and have graduated 5 baccalaureate students, 5 master degree students, and 5 PhD students. And he have received 2 grants and 3rd place in Caniforma 2012 and in the year 2019. He has also received scholarship of European Commission in the Netherlands, and so next slide please. And he has experience on clinical trials and some publications on pharmacokinetic studies in healthy volunteers, and he is also an expertise on HPLC methods. So with this, I cordially invite Dr. Carlos Hoyer sir to the session. Please welcome, sir.
Yes, thank you very much. Thank you very much. Just a second, I have to share my screen.
Sir, you can share, sir.
Excuse me, I have to share my screen.
Yes, sir. You can share your screen, sir. I already stopped sharing screen, sir. You can now share your screen.
Yeah. Okay, I will share my screen.
Okay, sir.
Yeah, here it is.
Yes.
Okay, so we start now with nifedipine. Yeah, here it is. Can you see now?
Yes, sir. It is visible, sir.
Okay, here this is the beginning.
Okay.
So I will speak about population pharmacokinetics of nifedipine in Mexican subjects. Okay, there are relationship between pharmacokinetic and pharmacodynamic that are done in order to get therapeutic information versus plasma levels. But it is possible to fit other effects with the drug levels. Excuse me, this screen is in Spanish, but you have the circles in blue show the plasma levels of a drug. So you have above the gray line, you have the therapeutic levels. All this green arrow shows the therapeutic levels. Above the red line, you can have adverse reactions. So the focus of pharmacokinetics is to have the most of the time in the green area. Nifedipine pharmacokinetics has been extensively studied. There are studies for different populations, especially Caucasian populations, and also some studies in Oriental populations. In Caucasian populations, it has been described an interaction with grapefruit juice. Grapefruit juice produces an increase in the levels of nifedipine. There are also other studies studying renal failure. And in other studies, the sublingual administration has been studied. This is a classical administration of nifedipine. In Mexicans, we have found a prevalence of slow metabolizers. That means that Mexican subjects have a slow metabolism of nifedipine. There are other interactions, drug-drug interactions with quinidine, cimetidine, and other drugs. This is the structure of nifedipine. Nifedipine is a dihydropyridine. It has two rings, and it is metabolized losing having the metabolite 1, losing the hydrogen here. This hydrogen is loose. It proceeds to other metabolites. One of the first studies on pharmacokinetics of nifedipine was done in the year 1983 by Foster. They administered intravenously 1 milligram, having a half-life of 1.77 hours. In other examples, REMS, also in the same year, 83, they administer oral administration of 10 milligrams of nifedipine. This is in a classic gelatin capsule. They found a bioavailability of 56% to 77%, with a terminal half-life of 1 hour, 0.26 hours. REMS is a study done in Germany. This is an example of 3 administrations of nifedipine. In the far left, you have the intravenous administration having a fast decrease and then a slower slope. Next is the gelatin administration with one compartment pharmacokinetics that arises very fast and then has one compartment in the oral administration. And the next slide shows the profile with a slow release nifedipine. So you have a lower, actually this slope actually is the absorption. The absorption determines this slope, because this is a slow administration, a slow tablet. So in this case, this is an example of flip-flop. This slope is the speed of the elimination on the right, on the left side. Okay. This is data of Mexican subjects. This is the average of the Mexican subjects for the capsule to up to 8 hours. So it has a fast absorption, then it has a slope for distribution and another slope for elimination. In the case of intravenous administration, this is a study done in the Netherlands by Dr. Klein Blusen. So he found a half-life lambda 1 of 0.09 and the final elimination of 1.7 hours. For the oral administration, they gave two capsules of 10 milligrams, and they found an absorption constant of 0.05. In this case, they had a lag time. That is a retardation time of 12 minutes, having a maximal concentration of 116 nanograms per ml. The bioavailability was 56%. The absorption constant was 4 minutes to the power of minus 1. and the maximal concentration was 25.9. Excuse me, this is for all this data is for the slow release. The left side shows the capsule on the left side. And in the right side is the data for the slow release. This is the plot for plasma after the capsule administration in healthy volunteers. This is data for Mexican subjects. In the open circles is shown 6 male. In closed circles, the data for 6 female subjects. This is our data. This data is from us. So in this case, we have almost only one compartment. Here, we show you the area under the curve distribution for the Mexican subjects in the state of Michoacán. That is a part of Mexico. So on the left side, we see a minority group for the fast metabolizers. The fast metabolizers have a small area under the curve. On the right side, we see that most of the subjects are slow metabolizers. Above the histogram, we have a probit conversion of the distribution. That is a probability unit. So for the first part, is shown in these open circles. Here, shown here. It is a line because this is a normal distribution. So this line is associated to this normal distribution. Then we have a broken line here corresponding to the anti-mode over there. And the other group has another slope, a different slope. On the upper side with closed circles is shown the probit curve from data in the Netherlands. In the Netherlands, they observe other situation with a majority of the subjects having a fast metabolism. So this data is bigger. And they have a smaller line corresponding to this group, but they have less subjects, but interesting, the same anti-mode. The anti-mode is located in the same place. So the closed circles correspond to the data of Klein Blusen made in the Netherlands. Okay. Here. So here is a comparison of our study. Well, actually, we did 2 studies. This study, Mexican 12, reference 12, is done with the capsule. In that case, we got a maximal concentration of 145. done in 12 subjects. In this part of the table, we show data of 6 subjects receiving the slow release nifedipine. The area was lower. And then we compare our data with 2 studies done in England. They have a smaller area. Also, in United States, they have a smaller area. And in German, they have a smaller area than our data. No, excuse me. I made a mistake. Sorry. This is our group of Mexican subjects, of 12 Mexican subjects receiving the capsule. Below are 6 subjects receiving also the capsule, but this is from a group of the Social Security. They receiving also the capsule of 10 milligrams. Capsule of 10 milligrams. So this is also in Mexican subjects. They have a smaller area than our study. Okay. Well, now we have to speak a little about the Mexican population. The Mexican population is an admixture of the Native American populating Mexico before the Spaniards arrived to Mexico, no? So this Native American, they come from this lake. This is the lake of Baikal that is in Russia. Now this is in Russia. At that time, this place was populated with people from China that were located very close to this lake. So they started the migration to the north of Russia, going to Siberia. They were walking, and then they crossed the Bering Strait, maybe chasing mammoths. So they arrived to Alaska, to America. After arriving to the north of the continent, they continue walking to the south until they reached Argentina, to the place of land of fire in the south of Argentina. This migration took about 1,000 years. All this migration of walking all the continent of America. There were another migration coming from this part of Russia, and this second migration arrived later. The first migration arrived 30 to 40,000 years ago, and the other migration arrived 10,000 years later. About 10,000 years ago. Just a second. Okay. The Mexican population have is a admixture of three components. The main component is the Native American component that comprises 65% of our genes. Then we have the Caucasian component of 30%, and we have also a component of African origin of 5% in average. This is data of average. During the colony, the admixture were a minority. We call them mestizo people. Even there were more Africans than Spaniards. They bring African slaves to Mexico. We had this admixture plot. This is a triangle with having one corner showing the component of the Native American in the top. In the bottom, we show the European component, the Caucasian component. And in this direction, to the left, is the component of the African. Each point shows one subject, each point. Okay. The north of Mexico has a greater Caucasian component, relatively greater Caucasian component. We have one state that is called Jalisco that got a very great French migration. We have done a study in this state, and we have found that in this case, most of the subjects are fast metabolizers. Okay, now we speak a little about the interaction of with grapefruit juice. This was discovered using felodipine. Felodipine is similar to nifedipine. So this study was done in Canada. They were making an interaction of felodipine with alcohol. They were giving the alcohol mixture with grapefruit juice. So they discovered that the grapefruit juice produced an increase in the concentration of felodipine. Actually, they perform, this grapefruit is given from a commercial for that you can dilute it and have the 1x concentration, but you can put more. It's a frozen preparation. You can put more water or less water. If you put less water, you have the normal concentration, but if you put less water, you have a more concentrated grapefruit juice, and in that case, you have greater effect on the concentration of felodipine. Well, now we make a small interruption showing you data in rats. Of midazolam. Midazolam is a classical drug for the cytochrome P450 3A4. In this case, we have here in the rats the concentration of midazolam here on the left side. And in the right side, we have the level of alcohol, ethanol, here, and the concentration of midazolam. The concentration is the same. It does not change because this is a small dose of ethanol. This is we call it social concentration of ethanol because you can drive with this concentration of ethanol. In the bottom, we have the effect of midazolam on the electrocardiogram, electroencephalogram. So this line is the control, and midazolam produces an increase in the frequency of beta frequency of the EEG. This is for midazolam alone, and this bigger plot is for the interaction with alcohol. So alcohol produces a greater effect of midazolam. And here we have the concentration of midazolam. On the right side is midazolam alone. In the left side is midazolam with ethanol. So the EC50 is moved to the left side. Okay, let's continue. Okay, the strategy is to adjust the concentration of the average by fitting all the data, so getting a population estimate of the parameters for one compartment or sometimes for two compartments. Okay, this is the example of the individual fitting. Then you have one concentration, and then you have the plot of the curve of the function. But for the population fitting, you have more subjects, and then you have another fitting different to the individual fitting. The hydropyridines are used in Mexico as an antihypertensive. In the world, they have longer half-life, like felodipine or amlodipine. The half-life with the capsule of gelatin is of 2 to 4 hours, depending on the population. Nifedipine is interesting because it was considered a prototype for cytochrome CYP3A4 and for its interaction with the grapefruit juice. This is the cytochrome 3A4. These cytochromes metabolize about half of all the drugs. In populations of Africa, it has been associated with the cancer of prostate cancer. Well, we have used the non-MEM program for the fitting of the pharmacokinetics. This program was developed by the University of California in San Francisco by Dr. Bennett and Dr. Bill. Non-MEM now is working with the company Icon. This is the golden method for pharmacokinetics. This is an example of a model for the pharmacokinetics using the exponentials for the administration, B for the elimination, this is for distribution. Alpha is for distribution, beta is for the elimination, and Ka is for the absorption. Here are examples of the fitting using non-mem for several subjects, no? So you can see the differences between the subjects. And then we have the histogram for the concentration. And this is the model for the interaction of alcohol with midazolam. And this is our results showing the blood pressure, diastolic pressure, systolic pressure. This is the cardiac frequency, and this is the respiratory frequency. Okay, with this, we finish the nifedipine. Okay, now I would like to show you the webpage of our Department of Pharmacology. This is in Spanish, but you can translate this webpage using the tool from Google. You can translate to English because our webpage everything is in Spanish. I showed you this to introduce our program of the PhD program. We have in this webpage, you can see the information about the PhD. You can come to Mexico, and here you can get a scholarship. So you can contact us in order to apply for the scholarship. Okay. Okay, now I will speak about the omeprazole. Omeprazole is an antacid that is widely used. It is metabolized by two cytochromes, 2C19 and 3A4. Here it is. Omeprazole is given as a racemic mixture because it has this sulfur. This sulfur can be R or S because of this. This is a chiral center. If you have the R molecule, it is metabolized by 3A4, producing sulfone, omeprazole sulfone, by 3A4. But if you have the S form, then it is metabolized by the C19, 2C19, giving origin to hydroxyomeprazole. Actually, there is a formula of only S-omeprazole that is called S-omeprazole. So in that case, it only produces hydroxyomeprazole. 3A4 is constitutively expressed in the liver, and it is also expressed in the gastrointestinal tract. It is the main responsible of all the drugs, of most of the drugs, about half of the drugs. 2C19 is responsible of about 10% of all the drugs. Mexico has now 126 million of people. I have mentioned that we have a tri-hybrid origin of American Indian, Caucasian, and African. Okay, we have a method for omeprazole that is reported in this paper. And this is the chromatograms. This is the blank chromatogram. This is an example of a standards. And this on the bottom, you have a patient, a volunteer who has received omeprazole orally. We had made a study using 122 volunteers coming from Jalisco, Sinaloa, and Nayarit. They firmed the informed consent, and they received 20 milligrams of Losec in fasting. One blood sample was taken at 3 hours after receiving omeprazole. And here is the results, the average. So we have the hydroxy metabolite in about 400 nanograms. Smaller is the sulfone, and greater is the omeprazole. This is the histogram of the ratio of omeprazole divided by hydroxyomeprazole. So this is the ratio for cytochrome 2C19. We have some subjects who were ultra-fast metabolizers. That means that they have a lot of hydroxyomeprazole. Most of the subjects are fast metabolizers, and we have a group of slow metabolizers. Or maybe we can call this poor metabolizers. This is for sulfone. In the case of sulfone, we have a similar plot, having also a group of people ultra extensive metabolizers. Most of the subjects are extensive metabolizers, and we have a small group of poor metabolizers. So the ultra extensive metabolizers have a very small ratio. We have about 2% for the subjects of 2C19. 22% were extensive metabolizers, and 6% were poor metabolizers for 2C19. In the case of 3A4, 7% were ultra extensive metabolizers, 83% were extensive metabolizers, and 11% were poor metabolizers. Okay, now we see another study for children receiving omeprazole. Babies receiving omeprazole because they had the problem of the reflux of gastroesophageal disease. This study was done in the Institute of Pediatrics Subjects Institute in Mexico City. Okay, the aim of this study was to determine the effect of omeprazole at 2 different doses given for 1 month on esophageal pH monitoring results. This was a prospective, longitudinal, and comparative study of 50 children under 1 year old. So these were babies. 22 children from the group 1 completed the study, and 21 from the group 2 completed the study. 3 children from both groups abandoned the study. Well, I think my time is finishing. So there was a significant improvement on the pH monitoring. The results after 1 month of treatment with both doses, with any statistical difference between them. Okay, with this, I finish my presentation. So if you have questions, I am open to receiving questions from you.
Thank you, sir. So thank you for your valuable presentations on nifedipine and omeprazole pharmacokinetics. You are explained about how that nifedipine and omeprazoles is undergoing on a mechanisms in a structural way, that what are all the changes are taking place in that structure, and how that it produce on a metabolite, and how that metabolism is differ from populations to on a populations, and how that other drugs can interact with that particular drug, and how it will modify that metabolisms, and interactions with on a food, particularly that nifedipine and phalodipine with that grapefruit juice. All those things you can clearly explain. Definitely it will be an eye-opening session for our all budding pharmacists and those who want to pursue their research on that pharmacokinetics. And there are some questions from our participants. One of our participants asked, is grapefruit juice interaction as well as grapefruit as such, whether both the things will produce a similar interaction or the interactions will be differ?
Grapefruit produces at some time they were blaming the naringenin. At the beginning, they were blaming the component of the grapefruit juice. The flavor of the grape is come from the a flavonoid that is naringenin. But later they show another components to be blamed for the effect of the grapefruit. The other components, they produce the increase. They block the action of the cytochrome P450-3A4. These components of the grapefruit, they block the metabolism by the 3A4.
Thank you, sir. There's another question from our delegates. That is Sarandi. Sir, will Russians also show similar pharmacokinetics to Mexicans?
Excuse me, please repeat me.
Sir, will Russians also show similar pharmacokinetics to Mexicans?
You mean the Russians?
Russians.
Sorry?
Will Russians also will have the same type of pharmacokinetics?
I do not understand.
Just a minute, sir. I will share in that screen and I will tell you.
No. The Russians, no. The Russians, they are Caucasians. Okay. They have a very fast metabolism. Compared to Mexicans, they have a faster metabolism.
Thank you, sir. There is another question from our delegates. Sir, as many people travel for different opportunities globally, and they no doubt adapt the environmental changes by time. Yes. Sir, do they adapt or change their drug metabolism within, which is actually dependent on their body response?
Yes. That's right. Yes, that is completely right. Yes, I agree with you. They can adapt. There is an adaptation. These differences are produced by adaptation with the environment. Our environment is very different from one place to another. Every environment produces in response. The body can adapt to, for instance, to the food. No, the main effect is given by the food. The food interacts with the liver. So in the liver, we have the main production of the cytochrome P450. So having different foods had a different action on the liver. Yes. So we have to adapt to our food. Yes, absolutely. Yes.
Thank you, sir. There's another question. What is the study design used for omeprazole pharmacokinetic studies?
We used we were based in previous information from the literature. In the literature, they show that measuring the concentration at 3 hours was okay to measure the effect on the metabolites of omeprazole. So we choose this time of 3 hours, and we administer the omeprazole. We took only one sample, only just one blood sample at 3 hours, because based on the literature. Yes.
Thank you, sir. The next question is: does omeprazole show different rate of metabolism in different races? Is there a need for dose adjustment for different rates of metabolism?
Yes. Actually, this family of cytochromes 2C have a lot of differences between races. We have a situation in the Oriental populations. They have more mutations. They have more mutations. Actually, we have two main two different routes of metabolism. I mean, we have two forms. We have two forms, two main mutations. on the 2C19. One is called asterisk 2 and the other is called asterisk 3. This asterisk 3 is only present in the Oriental populations. The asterisk 2 is greater in the Oriental populations compared to the Caucasians or the Africans. The Africans have less of the asterisk 2. So we have strong differences in the metabolism of omeprazole. Yes. So every population has to adjust the dose of the omeprazole. Yes.
Yes. Thank you, sir. Sir, what is the main criteria that differs the action of different drugs in different individuals?
Yes, the main criteria is to perform a pharmacokinetic study in every population. Every population has to do a pharmacokinetic profiling in order to know how they are metabolizing the drugs. So the main criteria is to measure the drugs using pharmacokinetics studies.
Thank you, sir. And any other participants, if they want to interact directly with the speakers, you can raise your hands. I will unmute it. You can directly interact with our speakers. Any other participants, if they want to directly interact with Dr. Carlos, you can raise your hand and I will unmute it and you can talk directly. Yes, Saran, Mr. Saran, you can unmute yourself. I gave a permission and you can directly interact. Mr. Saran D. Yes, you can ask your question, Mr. Saran. Please unmute your mic. Mr. Saran, you can unmute your mic and you can ask your question.
Hello, sir.
Yes, it is audible. You can ask.
Sir, Russians also originated, Mexico also originated here, Russia. But why why different metabolism, sir?
Sir, your audio was not clear. Can you ask once again, Mr. Saran?
Hello, sir.
Yes.
Sir, Mexicans also originated here, Russia. But why they differ in metabolism? Both have to be similar only, right?
Sir, I think Dr. Carlos.
So sorry, I cannot hear it properly.
Okay, sir. So he's telling that Mexicans are also moved from that.
Mr. Saran, just put your question on chat box so that he will understand and answer. Mr. Saran.
So he's asking that Mexicans are originated near Russia only, then why that metabolism is differing? Like that he's asking, sir.
And Dr. Carlos, is it audible?
I cannot hear the question.
Mexicans are also originated from Russia. Hello.
No, Mexicans are well, yes. That's right. They come from Russia.
Then
But
why do they have different metabolism pattern?
they come from Russia, but actually the population, it was a Chinese population. Actually, this was a Chinese population that has nothing to do with the Russians populations.
Okay, sir. So any more question, Mr. Saravanan?
No more questions, sir. We can conclude that session, sir.
I think with that questions are over, Dr. Carlos. Still, we will be getting some more questions from the YouTube live. All those we will be condensing and mail you. You please answer to the participants later. We can submit them. Now we can go for the that is certificate and memento presentation. Dr. Saravanan.
Yes, sir.
Okay, well.
Dr. Carlos, kindly accept our certificate of appreciation for spending your valuable time with us.
Yeah. Thank you very much. Bye-bye.
One more thing, we also have a memento, virtual memento. Kindly accept this also as a token of our appreciation for your valuable time sparing with us.
Yeah, yes.
Okay, thank you. Thanks a lot for your support, and definitely we will be in touch with you for your further academic and research collaborations with you.
Yes, teacher. Yes, professor.
Okay, thank you. Thanks a lot.
Bye-bye. Bye-bye.
Bye. So I think so with this morning session is completed. All the participants kindly join after lunch by 12:50, so that by 1 o'clock we'll be having a wonderful lecture from the expert on the pathology department. She is the head of pathology department. So hope that will also be one of the good session for the young scientists and pharmacists. So we'll break for lunch and we shall meet after lunch for the session with Dr. Janath. So we think with this, I think we can complete the morning session. We'll meet after the lunch by 1 o'clock. So we request you all to join by 12:50, so we can have the lunch session by 1 o'clock. We have to start the lesson session by in time, because all those who came from Mexico or this US, that time is entirely opposite. They are actually sacrificing their sleep and giving the lecture in the midnights. So let us not waste their time, and we'll join by 1 o'clock. We'll start by 1 o'clock the session without any more delay. And after 1 o'clock, we'll have the session, interactive, live interaction session with the international researchers by 2 o'clock. So thank you very much. Thank you all for joining us. We'll meet after lunch. Thank you. I think Dr. Saravanan, anything more to share with the participants?
Nothing, sir.
So we can sign up for now, and we'll meet post lunch session. Thank you.
Thank you, sir.
Thank you, sir.
Dr. Saravanan.
Sir, tell me, sir. Hello.
Dr. Janath joined.
Yes, sir. Yes, sir. Join, sir, doctor.
Audio connected?
Yes, sir. Connected, sir. Now it is connected, sir.
Okay.
Hi, good afternoon over there. Can you hear me?
Yes, madam. We are able to hear you, madam.
Great. Hi, everyone.
Welcome, Dr. Janath. I am the convener of the conference. On behalf of organizing committee, wholeheartedly we welcome you to this conference. So we'll wait for a few minutes and then start the session.
Okay.
Our students are joining in this Zoom or YouTube, Saravanan sir.
They're joining in YouTube, sir. Our students are.
Okay. Dr. Selvasankar, are you joined? Dr. Selvasankar.
Sir is joined, sir, already in the meeting.
Okay. So you can unmute him for some time,
Yes, sir.
Dr. Saravanan.
I unmuted him, sir. Already unmuted, sir.
So thank you, Dr. Janath. Dr. Selvasankar has given your contact and it has been materialized and we are here to this session. So Dr. Selvasankar was my student in undergraduation.
Okay.
And since then we had a good cordial relationship. That's why we are today meeting in this session. Thank you, Dr. Selvasankar. Dr. Saravanan, I think we can start slowly. So by the time we complete introduction, the audience will be joining.
Okay, sir.
Already our students are assembled in auditorium.
Okay, sir.
So we can start.
Okay, sir.
Dr. Sai Josna, madam, you can start.
Okay, sir. Good afternoon, one and all. I welcome you all for the second session on the International E-Conference on recent developments in biopharmaceutical sciences. In the morning, we had two sessions which were very helpful for us. And with the same enthusiasm, let us continue with the third session. Now I invite Mrs. Preeti ma'am to introduce our third session speaker. Madam, please, ma'am.
With immense pleasure, I would like to introduce Dr. Janet Pineda-Diaz, the head of the Department of Pathology at Hospital Angeles Levanstata, Mexico, where she is leading histological and cytological studies of patients. Dr. Janet completed her residency in pathological anatomy from National Autonomous University of Mexico. She procured her master's degree in genetic and molecular biology from the Center for Research and Advanced Studies of the National Polytechnic Institute. Dr. Janet did her general medicine at the Anocha University in Mexico. Her areas of research include clinical research for the treatment and prognosis of inflammatory, infectious, and autoimmune diseases and cancer. Histopathological evaluation of biomarkers in breast cancer, recurrent vulvovaginal candidiasis and disorders that predispose women to concurrent infections. Janet has publications in the areas of metastatic lobular carcinoma, splenectomy of solitary splenic metastasis in recurrent papillary thyroid cancer, intra-articular inflammatory myofibroblastic tumor, and ileal obstruction caused by transmural endometriosis. Welcoming Dr. Janet Pinet-Diaz to this session and requesting her talk on clinical and therapeutic use of biomarkers in breast cancer.
Okay, should I start sharing my screen?
Yes, you can start.
Okay. Okay, is everybody looking at my presentation now?
Yes, madam. It is visible, madam.
Great. Well, first of all, I want to thank you very much for the invitation. It's a bit past midnight over here, so I hope this will be worth it. I hope you will enjoy the talk and hopefully learn a bit about what I want to share with you. So as my presenter just said, I am a pathologist. I studied medicine and then I have been a pathologist for a couple of years now. And my most important interest right now is in cancer. And of course, one of the things that I am most passionate about is breast cancer, mainly because I am very interested in female health. And of course, as you know, this is a very important issue for us women. So this talk I'm going to share with you about the clinical and therapeutic use of biomarkers in breast cancer is mainly because I want you to understand how important it is in the clinical application for us as doctors, as medicine doctors, to have a good basic sciences research so we can use all of the information you guys provide for us and as a team being able to do good things for our patients. So this is a quick outline of the things I'm going to be talking about. First, I would like to talk about the epidemiology of breast cancer. I know you already know it is a big issue, but I want to highlight how important this disease is worldwide. About a bit of the risk factors, and then I'm going to try and explain to you about the histopathology of breast cancer. I think maybe some of you are familiarized with histology. If you're not, don't worry. I'll try my best to explain it to you. And then I'm going to talk about immunohistochemistry diagnosis of relevant biomarkers and the therapeutic and prognosis assessments we do as pathologists. So breast cancer is the most diagnosed cancer among women worldwide, and it accounts for 1 in 4 cancer cases. In 2020, 2.3 million new cases were diagnosed. This means that 1 in every 8 cancers diagnosed last year were breast cancer. But more importantly, during 2020, it caused almost 685,000 deaths, with an important number of these occurring in low resource settings. So breast cancer is a group of diseases in which cells in breast tissue divide uncontrolled, resulting in a lump or mass. Most breast cancer begin in the lobules, which is this part in this representation. So imagine if we took a bit of this tissue and look at it at more amplification. This is what we call the terminal duct lobular unit. These are the glands that basically produce milk. So what we are looking at here, can you see my arrow?
Yes, it's
I don't think
visible.
Is it? Oh,
Yes.
okay. Okay, so like I was saying, this would be the terminal lobular unit. This is the lobule, which is the part in which the milk is produced. And the ducts are these ones that are the ones that connect the lobules within each other, and also the ones that connect the lobules to the nipple. So remember this, because this is going to be an important part of the talk, the terminal duct lobular unit. About the epidemiology of breast cancer, this is a graphic that was published by the International Agency for Research on Cancer from the World Health Organization, and it was published on December 2020, so it is very recent. And as you can see, this graphic shows the number of new cases in 2020, both sexes, all ages, of all cancers. So I want you to look at this. Breast cancer accounted for almost 12% of all of the cancers that were diagnosed last year. This means breast cancer is the most common cancer diagnosed worldwide in both sexes, all ages. It is even more common than lung cancer, colorectal cancer, prostate cancer, stomach cancer, even more than cervix uteri, which was a big issue like in the last decade. Nowadays, it is not that common anymore because we have a lot of very accurate and early diagnosis tools for cervix cancer. So I want you to notice that breast cancer is the most important and the most common cancer of them all. And in this second graphic, which shows the number of deaths caused by cancer, also in both sexes, all ages, you can notice that breast is no longer the most common. It only causes around 7% of deaths. So lung cancer is the one that causes more deaths with 18%. Even colorectal is a more deadly cancer than breast cancer. This is because now we have very useful tools for the early and accurate diagnosis of breast cancer, as well as the chance to offer a personalized treatment for each patient, which can improve their prognosis. Still, the American Cancer Society establishes that approximately 1 in 8 women will be diagnosed with invasive breast cancer in their lifetime, and 1 in 39 women will die from breast cancer. So the thing is, breast cancer typically has no symptoms when the tumor is small, which is the moment in which the disease is more easily treated. So the screening for the detection of early breast cancer is very important. And as you might know, it is mainly done with a mammography, which is a kind of an X-ray study, which women should perform at least once a year once they are older than 4 years of age. And in fact, the incidence rates of breast cancer rose during the 1980s and the 1990s. And it was because that's when the mammography screening test was invented and started to be performed on women. So it doesn't actually mean that women didn't suffer or die from breast cancer before. It just means that in the 1980s, we started to be able to diagnose this disease and started to realize why these women were dying. And like I said, it is nowadays the most useful tool we have for the early detection of this disease. So there are a lot of risk factors for breast cancer, and one of the most important one is age. Breast cancer incidence and death rates increase with age until the 7th decade. Actually, the decrease in incidence rates that occur in women who are 80 years of age or older does not necessarily mean that they do not develop breast cancer. It might only mean that after the age of 80, less women undergo a mammography and less women are diagnosed. But the fact is that the older you get, the more likely you can develop breast cancer. In fact, the median age of diagnosis is 62.2 years. This means that half of women who developed breast cancer were 62 years old at the moment of the diagnosis or older. Another very important risk factor is the race or ethnicity. Incidence rates are highest among non-Hispanic whites, like you can see in this graphic, and it is this is the incidence, the pink, and as you can notice here, it is followed closely by non-Hispanic Black, but as you can see in the darker pink, non-Hispanic Black women have the highest breast cancer death rate. It is actually more than double than the Asian Pacific Islander women, as you can see when you compare these two bars. This is the lowest incidence in death rates, but the thing is non-Hispanic whites are the ones that suffer more from breast cancer. And there are another important risk factors of breast cancer that have been described, but these are mainly based on epidemiology and statistics. And some of them include alcohol consumption, early menarche, less than 11 years, that means the age in which you get your first period, excess body weight, high endogenous estrogen or testosterone levels, mainly before menopause. Also, late age at first full-term pregnancy, more than 30 years, late menopause, more than 55 years. So as you can see, they are almost always related with high levels of hormones. Never breastfed a child, no full-term pregnancies. Of course, one first-degree relative with breast cancer. The alterations in the genes are very important. We'll talk about that later. Obesity, it is also because of the high amount of estrogens that you have when you have obesity. Personal history of ovarian or endometrial cancer, physical inactivity, proliferative breast disease without atypia. These are like diseases of the breast that are not cancer, but that can predispose you to develop cancer. Recent and long-term use of menopausal hormone, recent hormonal contraceptive use, and weight gain in adulthood. So as you can see, the older you get, almost every woman can suffer from breast cancer. So now I'll talk to you about the histopathology of breast cancer. So this is a representation of the terminal ductal lobular unit I showed you before. So this is a drawing that represents a normal TDLU. So I want you to notice how it is made up of a single layer of cells. This is a drawing of the cells, and all of them look like very alike. And they rest in this membrane, that is the basement membrane. So when the cells acquire molecular changes that alter their reproduction rate, they become neoplastic cells, and they start forming masses within the duct. This is called carcinoma in situ. Carcinoma in situ basically means that the cancer is still within the terminal ductal lobular unit. And in this representation, the blue cells that, as you can see, are like very pleomorphic. They have different forms. They have different sizes of the nucleus. They are growing within the duct. But then, if these cells are able to go further and break the basement membrane, they become an invasive carcinoma. So try to remember this representation. And this is an actual microscopic photography of breast tissue. So this is normal breast tissue, and these are normal TDLUs. Each one of the tissue that you see inside a circle is one TDLU. And as you can see, try to remember this image, they look like very alike, very organized. This is a duct, this is a terminal duct, and this is an interlobular stroma. Remember, the stroma is basically, sorry, give me a second. The stroma is basically the tissue in which the terminal ductal units rest. And in this picture, we are looking at it a bit more closely. These are the terminal ductal units. These are the ducts. And now we are going to go even more closely. We are looking at one of the glands that form the terminal ductal unit. So these are normal cells. I want you to notice how these are. This is the nucleus, this is the cytoplasm. They look very alike, they are organized. It is a single layer of epithelial cells. Then we have these other cells that are called the myoepithelial cells. These cells are not epithelial cells, but cells that are around the epithelial cells and that give them like support. And then we have the basement membrane that you can see in a darker pink. So this is normal breast tissue. And this is carcinoma in situ. So compare the way it looks in here, how each of the glands is like very small with only one layer of epithelial cells. And in this one, look at how big each one of the terminal ductal units is. Look how much, how many cells are inside it. And if we go closer, look at these cells. Notice how they have different shapes of nucleus. They have different quantities of cytoplasm. Some of the cytoplasm is very clear. Some of the cytoplasm is very dark. Some of the nucleus are very dark. So these are all characteristics that pathologists look at to decide if this is a neoplastic cell or not. The more different the cells look between each other, then the more likely they are neoplastic cells. And this is an invasive form of carcinoma. In this one, notice that you no longer even are able to look at the form of the terminal ductal unit, the round form and the normal form. Now the cells are all over the stroma. They are invading even the adipose tissue, which is the one we look at in this part. We no longer are able to recognize glands. We only see a lot of individual cells. And this is a more amplification look of the cells. So you can see how different they are within each other. Like I said, with very irregular nucleus. We can even see some mitosis over here and over here. So when we look at an image like this, we are sure this is breast cancer. And since we no longer see basement membrane or myoepithelial cells, and we see how the cells are invading all of the stroma, we can accurately diagnose a breast cancer, an invasive form of the breast cancer. So after we diagnose the breast cancer, the most important part and the one I want to talk to you about in this talk is the immunohistochemistry, which is the technique pathologists use to be able to analyze important biomarkers for the accurate diagnosis of breast cancer. So the thing is, there are several treatment options that can help these patients depending on whether they have or not some alterations in different biomarkers. The three ones that we routinely analyze in breast cancer are the estrogen receptors, the progesterone receptors, and this other gene called HER2. So the hormone receptors, well, like we saw in previous slides, hormones are a big issue in breast cancer. If you have a lot of hormones, you are more likely to develop breast cancer. So this is why it is important to know if the patient has an alteration in the hormone receptors and then decide whether or not she's going to be able to receive a specific therapy. This is a photography of an immunohistochemistry assay. So basically in immunohistochemistry, what happens is we use an antibody that binds to a protein that we are looking specifically for. In this case, for example, we were looking for estrogen receptor. So remember, if you remember, for example, this image, you can see the cells in this stain that is hematoxylin and eosin, and it is a purple and pink stain. In this one, we only use hematoxylin, which is the blue stain that helps this other stain to be purple. So in this one, we only use the blue stain, and then the antibody that we use has a chromogen that in this case is brown. If this antibody binds to the protein that we are looking for, in this case, the estrogen receptor, then we will have a brown color. If we don't have a brown color, it means this tissue does not have the protein we were looking for. So estrogens and progesterone's receptors are actually transcription factor receptors, and they have an important role in development, growth, and differentiation of breast tissue. They are positive in 80% of the cases of breast cancer, and the important part and the reason we have to evaluate the presence of these receptors is because if the patient has it, if the case is positive, then the patient can receive a drug called tamoxifen that I'm sure some of you are familiarized with. To do this, we use the American Society of Clinical Oncology and College of American Pathologists guideline recommendations. These are the guidelines that we use worldwide. They were developed in 2010. And based on these ones, we have to report whether the patient is positive or negative for both estrogen and progesterone receptors. If they are negative, then tamoxifen is not going to have an effect. It's not going to help her. So it is not correct for the patient to receive this treatment. This is a nice example of estrogen receptors in which the result was positive. So you can see in the blue stain all of these neoplastic cells and how some of them are expressing the estrogen receptors, and so they are brown. This is a positive result, and this is a negative result. You can see only the blue stain, all of the neoplastic cells, but none of them has the brown color. So this means these cells no longer are expressing the estrogen receptor. Therefore, this patient would not be a good candidate for the tamoxifen therapy. This was reported as negative. This is another example in which I am showing you both estrogen receptors and progesterone receptors. This was also diagnosed as positive. And I want to show you how tricky this can be sometimes. I mean, the estrogen receptors is obviously positive. We see a lot of brown color in almost all of the neoplastic cells. But when we look at the other picture, then we see a lot of blue, not that much brown, but still, if you look closely, you can see some of these cells are still expressing the progesterone receptor. So this is considered positive as well. So this patient is a good candidate for tamoxifen. It is going to help her. And then we have this other biomarker, which is called HER2. HER2 is a gene that has an amplification in around 50% of invasive breast cancer. And it is a very important part of the prognosis of the patients because if they have an amplification of HER2, the disease is going to be more aggressive and the patient is going to have a worse prognosis. But there is a target therapy for HER2. So if the patient has an amplification, then she can receive this other therapy, which can which can give her a better prognosis. So just to clarify, the amplification of HER2 means that it has more number of copies of the gene than a normal cell. And to be able to analyze these, we would have to use a special technique, which is the fluorescent in situ hybridization. I'm pretty sure you guys are familiarized with it. If not, we will talk a bit about it. And the consequence of having an amplification of a gene, of course, is that we will have an overexpression of the protein. HER2 is a protein that expresses in the cell membrane. So if we have an amplification and we have more protein in the cell membrane, we will be able to analyze this as well with immunohistochemistry. We also have guidelines from the American Society of Clinical Oncology and the College of American Pathologists that we follow for the evaluation of HER2 in breast cancer. These guidelines were developed in 2018. So this is basically what happens with HER2. So HER2 is a protein that is a part of the HER family. The HER family is made of HER1, which is also called EGFR, which is a very important protein that is present in a lot of cancers, for example, lung cancer. There is also HER2, HER3, and HER4. So all of these are, like I said, membranes, proteins that express in the membrane, cell membrane, and they all have a tyrosine kinase domains. So the way they work is when they have a specific ligand that binds to them, they are able to dimerize. HER2 is able to dimerize with HER2 as well, but it can also dimerize with another member of its family. And once this happens, it produces a lot of signaling cascades that ultimately will cause cell proliferation, cell survival, cell mobility and investment. And it also activates other transcription factors that can even produce more blood vessels, which ultimately will give more substrates to the tumor cells. So it is like this cycle in which every single thing is making the cell to reproduce, to grow even more. And this is how HER2 produces, the role of HER2 in breast cancer. And we have this target therapy, like I told you, that is called trastuzumab. Trastuzumab is a monoclonal antibody that is used mainly to block this cascade of HER2, and it is able to stop the progression of cancer. So what happens is trastuzumab blocks dimerization. It binds to the dimerization unit of the protein of HER2, so it is no longer able to bind with another protein. Therefore, the cascade, the signaling cascade that starts the cell proliferation and growing and etc., it no longer happens. But also, trastuzumab is able to call some immune effector cells. This is going to activate antibody-dependent cell-mediated cytotoxicity. So in the end, it also helps to tumor cell lysis. So these are the two main ways trastuzumab works. It is a very good drug that helps a lot in the progression of cancer. And like I said, when the patient has an amplification of HER2, it has a much worse prognosis. So trastuzumab can help a lot in these cases. This is the algorithm that pathologists use to determine whether HER2 has an amplification or not. So the first approach we always have to use is also with immunohistochemistry, basically because it is easier and cheaper. So this is the algorithm we use. We have to evaluate the whole cell membrane, and we have to see whether it is positive or negative, whether it stains brown or not. You might remember that for example, in the estrogen receptor immunohistochemistry, we only look for a little dot of brown. This is because the estrogen receptor is in the nucleus of the cell. So of course, when the antibody in the immunohistochemistry binds to the estrogen receptor, it is going to happen in the nucleus, which is why we only see this small dot. But HER2 is a bit trickier. In HER2, we have to look for the stain in the cell membrane. This is a good example. I'm sure you can see how the nucleus is blue, but we can see how all of the membrane is brown. So this means this cell has an amplification of HER2. So like I was saying, if we see, like in the example I just showed you, a complete cell membrane strong stain in more than 10% of the cells that we are evaluating, it means, like in this example, I mean, look how brown, how dark the brown is. This is a positive case. We have to evaluate it with 3 positive, and it means this is a positive case. It means the patient has an amplification of HER2. On the other hand, if we absolutely see no signal, like in this case, there's no brown at all, then it is an absolutely negative case. If we only see incomplete cell membrane weak stain in more than 10% of the cells, like in this case, as you can see, we can see some brown, but it is not complete, not the whole membrane is brown. There are cells that are not brown at all. So this is also a negative case. But there is this other group, which is a much difficult group in which we see complete cell membrane, but it is weak in more than 10% of the cases, of the cells, sorry. For example, in this one, compare these 2 pictures. This is strong stain, this is weak stain. It is complete, we can see it all around the membrane, but it is very weak. This one is called an equivocal. I'm sorry, this was in Spanish originally. This is why we have this word. This is diagnosed as equivocal. And in this one, we have to do the FISH. So this is a good example of a positive HER2. You can notice that not all of these cells are positive, but the cells that are positive represent more than 10% of the cells we are evaluating, and they have very strong stain. So this is a positive case. On the other hand, this is a negative example. You can see some browning here, but it is less than 10% of the cells we are evaluating, and most of them are not even complete. It is a weak stain, it is incomplete, so it is a negative case. So what happens when we have an equivocal case? We have to do the FISH because we are not sure whether this patient is a good candidate for trastuzumab or not. So what happens with FISH? Like I was saying, in a normal cell, we only have 1 copy of the gene of HER2. HER2 is a gene that is located in the chromosome 17, in the long arm of chromosome 17. So the way this works is we use a probe that is directed towards the HER2 gene. And in this example, it is going to be a red signal. And we use another probe that is directed towards the centromere of this chromosome, of chromosome 17. It is called SEP17. And in this example, it is a green probe. This is the way we would look at in the FISH. We would see 2 green signals and 2 red signals. This would be like a normal cell, right? But if we have an amplification, if we have a lot of copies of the HER2 gene, then what happens is we only see 2 green signals, meaning we have 2 centromeres, but we see a lot of red signals. In this example, for example, we have 8 red signals. So what we do is, based on the immunohistochemistry, we are going to locate the section that has the most signals, that is the most positive part of the tissue. And in that part of the tissue, we are going to evaluate the fish. We have to count 20 cells, and we are going to look at the number of HER2 signals. In this example, we have 8. And the number of the centromer signals. In this example, we have 2. And then we have to have a ratio, an index. In this example, it would be 4. And this is the algorithm we have for the fish. So basically, when the index is 2 or more, we have a positive case. But if the signal is 2 or is less than 2, then we have a negative case. There are some cases in which not even the fish can accurately tell us if HER2 is amplified. Sometimes the ratio is 1.9. So there are some tricky cases in which not even the fish can help us, but most of the time it helps us around around 0.5% of the cases are the ones that we cannot conclude even with fish. These are actual photographies of a fish. So this is a normal cell. And as you can see, we have 2 red signals and 2 green signals. So there is no amplification of HER2 in this example. And these are 2 cells that do have the amplification. So we have these 2 signals and 1, 2, 3, 4, 5, I think 6 red signals, meaning HER2 is amplified. And in this one, we have a lot of red signals. So this is the way we analyze and evaluate the fish. But why is it so important for us to accurately diagnose the amplification of HER2? Mainly because trastuzumab is a very harsh medicine for our body. It has a lot of toxicity, and these are some of the symptoms you can develop, like fever, nausea, vomiting, diarrhea, infections, headache, fatigue, rash, alterations in your blood cells, anemia, a lot of pain in your muscles. So it is not a good idea to be taking this medicine if you don't really need it, because it is not going to help you and it's going to give you a lot of secondary effects. This is the trastuzumab, it's called Herceptin. And this is another big issue, the price of Herceptin. A full 1-year course of Herceptin treatment costs about $70,000. And there are some patients that have to take Herceptin for around 5 years. So of course it is very expensive, and not all of the patients are able to afford it. And it is also a public health issue. So it is very important to have an accurate diagnosis of HER2 and determine whether the patient is going to have real benefits from using it or not. And this is almost the end of the talk. There are other tests that are a bit newer that can also assess the molecular state of breast cancer. This is the most popular one. It's called Oncotype. So Oncotype is a 21 genes test, and it helps oncologists to decide whether chemotherapy is going to benefit the patients. And more importantly, it helps to realize if the patient has a high risk of recurrence. So as you know, there are some genes, for example, BRCA, that if you have this gene mutated, then you will probably develop breast cancer, bilateral breast cancer, and you can also develop other kinds of tumors, for example, ovarian tumors. So these kind of tests are able to see if you have these alterations or not. And they help a lot the oncologists to determine the best way of treatment. These are the 21 genes that Oncotype measures. So these are genes basically for proliferation, for invasion, of course, HER2, estrogen, and some other genes that have shown to be a part of the pathogenesis of breast cancer. So, and well, of course, it uses 16 cancer genes and 5 references, not that 21 are related with breast cancer. The way it works is with a real-time PCR, and then they have a whole algorithm with biostatistics, which is not public yet. So we don't know exactly how they determine the risk assessment, but they give you back a report in which you are able to know if you have an alteration of one of these genes. And if you do, what is the risk of recurrence, and if you should undergo like maybe a surgery or some kind of treatment that is going to allow you to avoid recurrence. The most important part of oncotype diagnosis is that it has to be at an early stage. It means if you have a very advanced disease in which you already have metastasis, then it is not going to help you that much, because if you have metastasis, you already have a bad prognosis, and this assessment is not really that important. So this is basically what I wanted to show you. I have some closing remarks. So like we saw, breast cancer is the most frequent cancer in women worldwide. The early detection can improve its prognosis, which is very important. And that is why the undergoing mammographies once a year, once you are 40 years or older, is very important, even if you don't have a family history of cancer. Because as we saw, there are some risk factors that are not related to your family history. Pathological examination can determine personalized therapy. Nowadays, every single breast cancer has to undergo immunohistochemistry for these 3 biomarkers. You cannot have any kind of therapy other than surgery, of course, if you don't have this pathologic assessment. And immunohistochemistry is the best tool pathologists have to accurately evaluate the molecular target therapy. Like I said, there are some other tools, but immunohistochemistry is the easiest, the most straightforward one. So this is the one we usually use. And there are some other tools, including in situ hybridization and multi-gene expression tests, that can give further insights of the pathogenesis of this disease. So basically what I want you to know with this talk is that breast cancer, as well as almost every single cancer, is a very complex disease. There are a lot of genes that are involved in the pathogenesis of this disease. The good thing about breast cancer is right now we have a lot of tools to determine whether these genes are altered or not. And the most important part is that the therapy we have for breast cancer right now can be personalized. Meaning, if you know what genes you have altered and which ones you don't have altered, then you are able to decide which therapy is going to be the best one for you, and that can improve your prognosis a lot, which is why I think breast cancer, even though it's the most common cancer, It is not as deadly as, for example, lung cancer, in which we still don't have a lot of tools to determine the best therapy for the patients. And this is a photography of the city I live in, Mexico City. This is Paseo de la Reforma. This is almost downtown Mexico City. And this is a monument called Ángel de la Independencia, the Angel of Independency. And it is a monument that commemorates the independence of Mexico from Spain, which happened in 1810. This is a very nice picture because during Sundays, this street, which is usually very crowded with a lot of cars, is closed. So you can ride your bikes and you can go skating with your family, but this is not the way it usually looks. So this is it. I would like to know if you have some questions.
Thank you, madam. Thank you for your wonderful session on clinical and therapeutic use of biomarkers in breast cancers. On behalf of that organizer, first I would like to big thanks for you, madam, because we know that it is a time is 2:10 a.m. in this time. That is an early morning. You are given a very wonderful lecture to our budding pharmacist. There is a
Thank you.
Our participants. What are all the hormonal changes may lead to breast cancer?
I'm sorry. Can you repeat the questions? Do you know if I can see the questions as well?
Yes, it was from YouTube, madam. That we
from YouTube. Okay.
What are the hormonal changes may leads to breast cancer?
The hormonal changes that lead to breast cancer. Breast cancer. Like I said, the most important risk factor regarding hormones is high levels of hormones. That is why some of the risk factors that I described, if you look at them, most of them are related to high levels of estrogen during long periods. For example, if you start your periods when you are very young and have reached menopause when you are very old, that means you will be exposed to estrogens for a longer period than a woman that starts her period older and starts menopause younger. Another important risk factor is if you don't have pregnancies. So that is also because when you're pregnant, the estrogen levels decrease a bit because you have higher levels of progesterone. So that is like a short period, a 9-month period in which you are not exposed to high levels of estrogen, and it is somehow protective of breast cancer. And you might know, you might remember that I think in the 1980s maybe, there was a lot of talk about how hormonal contraceptive pills seem to be a risk factor for developing breast cancer. That was because in the beginning, when they were first developed, the contraceptive pills had a lot of estrogen. So it was demonstrated that yeah, this high level of estrogens could help the development of breast cancer. So nowadays, as you guys might know better than me, the contraceptive pills have much smaller amounts of estrogen, and I think there are even newer treatments in which they only use progesterone, not even a bit of estrogen. So the thing is, the longer and higher levels of estrogen you are exposed to, the more likely you can develop breast cancer. Of course, breast cancer, as any other cancer, is a multifactorial disease. It is not only one thing that causes it. You have to have a lot of other things that together cause the disease. But yes, high levels of estrogen is a very established risk for breast cancer.
Thank you, madam. There is another question. Is there any risk if taken herbal supplements during treatment?
I'm sorry. Is there any risk if what?
If we taken herbal supplements during treatment.
No, I'm sorry, I didn't get the question.
Is there any
risk? Sorry, can I explain to her, doctor?
Yes, yes.
Yeah, if they take any kind of medicinal plant related treatment, Janet.
Ah, okay. To be honest, I have no idea, but I can show you. Let me see if I'm going to show you a very interesting case I saw. I have to get out of this. I haven't met anyone with herbal or like alternative use of other treatments, but I did have a patient that was undergoing a ozone therapy. I don't know if any of you have heard of that one. I think it is based on like high levels of ozone, and they are supposed to like decrease the levels of oxygen that the neoplastic cells get. So hypothetically, they can like reduce the reproductive rate of the cells. So you have to look at this. I hope you guys don't have a stomachache after looking at this image. Let's see. This patient was very young. She was diagnosed with, I think it is this one. She was diagnosed, I think, like at 42 with breast cancer. She didn't Can you look at it? Are you looking at my slide right now?
Yes, ma'am.
Yes, okay. So this is in Spanish, sorry. She was 44 years old and she was diagnosed with breast cancer, and she decided not to undergo chemotherapy. She didn't even undergo surgery, and she decided to go to this alternative therapy. And this is what happened. This is a mastectomy. So the breast was underwent surgery, it was taken off, and this is the nipple. All of this is a tumor. So the thing is, I cannot for sure say that this alternative therapy was the cause of her tumor growing so much. Maybe she just had a very aggressive kind of cancer. And since she didn't have any treatment, this is what happened in a period of 4 months. I cannot say for sure, but the thing is, this is not a common breast cancer. This is a very aggressive one. And I don't know if I have this is another picture. So, I mean, look, this is a rule, right? It measured almost 10 centimeters. It was very big. It was very aggressive. It went out of the skin. It was a terrible case. I don't think I have the microscopic image, but it was, the cells looked horrible. They were very atypical with a lot of mitosis. So it was a terrible case. And after 6 months, the patient died. So I'm not sure I am answering the question. I don't know if herbal or other alternative therapy can help, but I do think it is very important to, if you want to try that treatment, go ahead, but do not abandon completely the traditional well-established treatments. We know these treatments work, so you can try other things, but I wouldn't recommend anyone to completely abandon them and undergo alternative therapy.
Thank you, madam. That another question is there any chance to get breast cancer in the children?
Yeah, it is not common. but it is possible. When you have a germline mutation in some genes, including a gene called BRCA, it is possible for young women to have breast cancer. And interestingly, in the past few years, there has been an increase in the incidence of breast cancer in younger women. Like I said in the first couple of slides, the median age of diagnosis right now is around 60 years old, but we are looking at younger women with diagnosis. This case I just showed you, she was 40 years old. Last week, I saw a case of a patient who was 36 years old, which is my age, by the way. So younger women are starting to get diagnosed. Is it because we are now able to diagnose the disease earlier? Maybe. Is it because we have now a lot of environmental factors that are affecting us and causing more cancer at earlier stages? Maybe. We are not sure about that. But yeah, young women can also have, children can also have breast cancer. And of course, males can also have breast cancer. It is not that common, but I have had at least 4 cases of male breast cancer. In those cases, the diagnosis is almost identical to the breast cancer in females. We have to do the same tests. We have to assess, of course, estrogen receptors, progesterone receptors, HER2, and of course, the surgery is the most important part of the treatment.
Thank you, madam. Then another question is that normal cell also express progesterone and estrogen receptors. Is there any chance for misinterpretation?
Oh, okay, that's a good one. Yes, the normal cells express these receptors. So the thing is, if you do this stain to normal tissue, it is going to be positive. The important part is that you evaluate it in the neoplastic cells. If the neoplastic cells are positive, then it means the drug tamoxifen is going to affect the neoplastic cells, and it is going to help for the treatment of the breast cancer. But sometimes in the same slide, you have both neoplastic cells and normal cells. And it is actually like a positive internal control. You can look at the positivity in the normal cells, and it means your stain was done correctly. It means you can trust your stain because they are always going to be positive. If you see normal cells that are not positive for the estrogen receptor, then it means your stain isn't good. So the important thing is that you evaluate only the neoplastic cells. I hope I was clear with my answer.
Yes, madam. Thank you, madam. Still, we are receiving many questions, madam, but we will send these things to your email ID and we can forward that answer to that.
Yeah, of course. Okay, can I answer one really quickly? This one that says, what's the stage of the breast cancer? Okay, what I was referring to when I said stage, we have this way of evaluating cancer in general, that is saying the stage of the cancer. Basically, it means whether the cancer is only contained in the organ that it originated in, or if it has already metastasized to another organ, meaning the neoplastic cells are able to travel via the blood vessels or the lymphatic vessels and affect other organs. The organs that are most commonly affected from metastasis in breast cancer are brain and liver. So the stage is what you decide based on how big the tumor is, whether it has or not affected the skin of the breast, and whether it has or not metastasized to another organ. When it has an advanced stage, then the thing is the prognosis is bad, and sometimes they don't even, the oncologists don't even ask for this whole evaluation because they are no longer able to offer the treatment for the patient.
Yes. Thank you, madam.
Yeah, sure.
Thank you for your nice session and answering for all the queries which was raised by all our participants. So now I am requesting our principal sir to give a conclusion. Principal sir.
Yes, Dr. Saravanan, I am here.
Yes, sir. You can conclude that session, sir.
Thank you, Dr. Janath, for joining us and enlightening about the breast cancer. That many of the pharmacy students and the other biologists also take this as one of the important topic in their career. You have enlightened very well in detail, and the way you have answered the queries really very much appreciable. And I thank on behalf of all the audience that you are speaking in the midnight, but it's not looking like that. We are talking with the full of energy. Thanks a lot for sparing your valuable time. And definitely in the coming days, we'll explore organization, and we have long way to go our relation. Thanks a lot for sparing your valuable time. Kindly accept our memento, virtual memento, and the e-certificate as a token of our appreciation for your contribution. Kindly accept it.
Thank you. Thank you so much.
Okay.
Thank you for having me.
Thank you very much. Bye-bye. So next we'll be having another session. If you are free or if you are able, you can stay for some more time. Otherwise, you can have already, it's midnight. Thank you very much. Thanks a lot.
Thank you, ma'am.
Okay. Madam, Sai Josna, madam, you can go for the next session.
Yes, sir. So now we have a session for live interaction with the international researchers. So now I invite Ramesh sir, Associate Professor, Department of Pharmacology, to introduce our international researchers. Ramesh sir.
Ramesh sir, you can unmute yourself and you can talk, sir.
Good afternoon, all. First, I would like to thank our chairman, sir, M.N. Raju Garu, and vice chairman, Ravi Verma Garu, and our beloved principal, Alagar Swami, and staff and dear students. Very good afternoon for this e-session on biopharmaceutical recent trends in biopharmaceutical sciences. And here we have live interaction session with three researchers, Dr. Shalva Kumar sir, and Dr. Vishnu Nayak, and Mr. Jagan Mohan. First, I'd like to go with Dr. Shalva Kumar, brief introduction. So Dr. Shalva Kumar here currently working with a postdoc in Sidra Medicine, Qatar. And he has also done another postdoc in Science Tau Institution, Mexico, 2017. And he has obtained PhD in Mexico from the same Science Tau in 2015. And he has done his master's in NIPER, and he completed his Bachelor of Pharmacy from SB College of Pharmacy, Shivakashi, Tamil Nadu. And his areas of research are his expertise in the field of human microbiota and the microbiome and its association with the host metabolism and dysbiosis in relation with other metabolic and autoimmune disorders. And he has been part of 6 book publications, and also he published both national and international publications, more than 37. And he attended around 40 conferences, and he has more than 10 years experience. And it's my immense pleasure to invite Dr. Shalva Kumar sir to our live interaction session. And we go with the other speaker, Dr. Vishnu Nayak. Dr. Vishnu Nayak So he has done his MS, PhD, and postdoc in Department of Medicinal Chemistry. So currently he is with Hebrew University, Israel, and he was doing his postdoc. And earlier he was with other university, Chulalongkorn University. In 2018, he has also obtained another postdoc. And he also from in India, IIT BHU also has done another postdoc. And he has been part of a research assistant, Central Drug Research Institute, India. And he obtained PhD from BITS Ranchi in 2016. And he has done his MS from Naipur, Uttar Pradesh in 2011. And he is our college student, MNR College of Pharmacy in 2009. And his areas of interest are organic and synthetic medicinal chemistry, nanotechnology, and molecular biology. And he also part of 3 book publications, and he published both national and international more than 34 publications. And also he was received awards and fellowship from BITS, NIPER, and post-metric fellowship as well. And he attended more than 11 conferences, and he also have more than 10 years experience both in research and academic. Thank you, sir. We heartily welcome you for this online interaction session. Next, we go with the other speaker, Jagan Mohan Garu. Jagan Mohan Kaipa, he also done MS in biotechnology from Martin Luther University, Germany. And currently, he is pursuing PhD from Helmholtz Center for Infection Research from Germany. And he is also our college student, MNR College of Pharmacy. And his areas of interest, biotechnology, and he's also part of both national and international publications, more than 3, attended 5 conferences, and he has more than 5 years experience, and he's currently pursuing PhD in Helmholtz Center for Research and Infection. And I heartily welcome you, sir, for this live interaction today's session. Thank you. So with this, we can go with the online interaction. Here we have variety of questions you can ask concerned with the research or postdoc. So what's kind of questions you have? Both teachers and students, both you can raise your hand, interact with our live speakers, Dr. Shalva Shankar, Mr. Jagan Mohan, and Vishnu Naik. Thank you, sir.
First, I would like to welcome all the speakers. Now I am requesting Dr. M. Selvasankar to give his words to that our all gatherings.
Thank you, Dr. Saravanan, sir. Yeah, this conference has been organized by our Dr. Alagaswamy, Principal of MNR College of Pharmacy. It's a wonderful experience. I hope all the audience, they enjoyed the talk given by all the international speakers. And this is one of such a great platform for the B-pharmacist and those who are in pursuing master degree. It's a wonderful experience, even though in this Corona period, they are actively organizing many conferences. During our period, it's quite difficult to attend different conferences in different cities. Now, because of latest development technologies, we can attend many sessions by staying in home. So I hope those who are coming today to attend this session, they can come up with any question, whatever they related to research, or it is related to if they have any queries regarding their career, we are happy to attend. And I welcome all the audience once again for the session. Thank you very much for having us here.
Thank you, sir. Now I am also requesting Dr. Vishnu Naik to give his opening remarks. Sir, Vishnu, sir. Dr. Vishnu Naik.
Dr. Vishnu Naik. No problem, Dr. Saravanan. Even Mr. Jagan Mohan also, due to extreme harsh climate in Germany, he will be joining little late, maybe in 5 or 10 minutes.
Okay.
We can start with the session. So Dr. Selva Shankar will be addressing the queries of the students. So now we request the participants to interact with the international researchers. Now Dr. Selva Shankar is ready to answer your questions.
Those who want to ask questions, please raise your hands. I will unmute it. You can directly interact with our researchers, international researchers. Please raise your hands. I will unmute your mic. You can directly interact with our international researchers. Sir, Selvasankar sir, there is a general questions from all the participants, so that if they want to pursue their research in that abroad, what are all the preliminary things they have to do when they are doing their UG programs? Sir, please unmute your mic and talk, sir. Dr. Selvasankar sir, please unmute your mic, sir.
Okay. Yeah, it depends whether you want to join for masters or the PhD. Most of the time in Europe, they will ask for, apart from your normal marks in your grades, you have to go for GRE or TOEFL. And also many countries like Latin American countries, including Mexico, Argentina, Brazil. And in these countries, they don't ask any of this grade. So directly with your master's or your PhD, you can apply directly for that. You can approach your scientist of interest. You can directly approach them. Then by normal interview, by presenting your previous thesis work, they can directly give you the acceptance letter. And in US universities, most of the universities, they will ask for TOEFL. So based on TOEFL score, then you can directly apply to any particular professors or directly to the universities. I think, did I answer the question, sir?
Yes, sir. Yes, sir.
I think
And
that's
what are all the opportunities for a pharmacist in your country, sir, in the Qatar? In the Qatar, what are all the opportunities?
In Qatar, for example, honestly say, after my bachelor's, I didn't practice much the pharmacy field. Mostly my master's also belonging to biotechnology. So slowly I was focusing more towards the research, mainly on human microbiome field. I'm not exactly practicing in the pharma field, but mainly in Arab countries, the most common job people here they come for as a hospital pharmacist. For hospital pharmacists here in Qatar, first they have to pass the exams to get their license here. So every year they have to maintain certain points to maintain their license so that they can practice as a pharmacist. So the main job here is to work as a hospital pharmacist. Here there are no big pharmaceutical industries, very few industries, but the jobs are very limited. So, and apart from that, research field. Here in research, I don't recommend here in Arab countries to directly come for a PhD opportunity, because even though you get a PhD, but here the scholarship is less. You have to, apart from that, you have to pay the tuition fee, which is expensive compared to other countries. But as a researcher, you can directly join here. You will have good facilities here. And for pharmacists, the main job here is hospital pharmacist, and then you can find some industry jobs.
Thank Thank you,
Sir,
sir.
is there any fellowships available for graduate and postgraduate students in abroad?
Yeah, I have been in Mexico for almost 7 years. Yes, they do have fellowship for both masters, PhD, and even for postdoc. Yes, that amount, if you expect like their Mexico, their currency is like 4 times more than our Indian rupees. So the whatever this fellowship you earn, it's more than enough for your stay, including all your living expenses there, and you can save some money. And it's a very good atmosphere. I recommend you can approach. In other Europe countries, yes, everyone they offer scholarship for their masters and PhD. But I think in Mexico, even they don't even charge you the tuition fee. Just all your scholarship fee mainly you can. That is mainly for your living expenses in Mexico. In other countries, I'm not sure about it. Maybe there may be a certain amount you have to pay for the tuition fee for your each semester, I guess.
Okay, sir. Thank you, sir.
Yeah.
Dr. Saravanan, Dr. Vishnu Naik has joined. Dr. Vishnu Naik? Dr. Vishnu Naik? Kindly unmute and say a few words as an opening remark. Or say a few words about your research background.
Sir, again, sir is disconnected, sir.
Okay, then we'll go ahead with the questions and Dr. Sindhu Sangha will be addressing.
Yes.
Yes, Dr. Saravanan, continue.
Okay, please.
Sir, good afternoon, sir. Sir, good afternoon, sir.
Hi, good afternoon.
Sir, I would like to know how to avoid plagiarism while writing thesis or research papers. This is my first question. And second question is, so how to do PDF in abroad? So what are the sources and requirements, sir, in abroad after completion of my PhD?
Yeah. So my first question, I will answer to your first question. So to avoid plagiarism, just do your original research and don't copy your work from other things. And if you copy also, almost all the editorial journals, they have particular programs which can easily identify when we do copy paste any stuff. So always write on your own words. Don't worry about whether we are writing English or not. When we have clarity in our concept, there won't be any need to do the copy paste thing. So maybe I think that answer your
Sir,
question.
especially in chemistry, so many structural similarities there, sir. So that is the problem for plagiarism. So how can any tips in chemistry for avoiding plagiarism?
I think maybe our principal, Dr. Alagasamy, is the best person to answer this question since he's the eminent scientist in this. So maybe I will invite you, sir.
So, one of our other speaker is also joined. So I heartily welcome Mr. Jagan Mohan to this live interaction with that international researcher. Sir Jagan Mohan, sir, please give your opening remarks to our gatherings. Sir, please unmute your mic and you can talk, sir.
So, am I audible to everyone now?
Yes, sir, it is audible, sir.
Thank you so much. So, first of all, hello everyone, and my apologies for the late joining because the weathers were extremely cold and it was snowing like crazy today in the morning. So it took some while for me to drive home. Thanks for everyone for inviting me. So to introduce myself, I'm Jagan Mohan Kaipa. So I was the former student from MNR College of Pharmacy. I was the batch for 2007 to 2011. Then I came to Germany to do my master's in pharmaceutical biotechnology in 2014, and then I joined as a research assistant for 3 years in BioNTech. I think we have heard this company since last few months because of the new COVID vaccinations which they have developed. And then after that, I started doing my PhD at Helmholtz Center for Infection Research in biotechnology for membrane proteins.
Thank you, sir. Once again, on behalf of organizing committee, we are welcoming you for this international e-conference, sir.
Thank you so much.
Now that participants can ask their questions. Mrs. Pratibha.
Good afternoon, sir. Sir, good afternoon, sir.
Yes, good afternoon, madam.
Good afternoon, sir. Sir, my question regarding the antiviral compounds from herbs, sir. So, is there any available database? No database is there for antiviral compounds, sir, especially with respect to the herbal drugs. Any antiviral compounds from the herbs we are getting, no, sir. Is there any database? Known database is there, sir, for searching the literature?
Dr. Vishnu Nayak. Dr. Vishnu Nayak is working on this antiviral compound, that COVID-19 only. I think he can answer better. That's why I'm waiting for his reply. I think due to some technical issues, he is joining lately. We'll wait for a while, madam, so that you can listen from him. Otherwise, I'll answer later if he don't join. Maybe we'll go for the next question, Dr. Saran.
Mr. Sai Chaitanya.
Sir, good afternoon, sir. My question is to Dr. Selvasenkar, sir. Sir, your research was about the microbiome therapy. So I would like to know what are the possible uses of the microbiome therapy in which kind of diseases, and what are the benefits of this therapy compared to the pharmacologic treatment?
Yeah, that's a very good question. So, when I started my PhD work, the main aim during that time, is there any particular microbe which resides in our human intestine, which is mainly related, apart from our normal digestive system, these microorganisms also doing the extra work by doing the substrate which cannot be digested by our human digestive system. They will take as a substrate and produce extra metabolites that will have an impact in our human health. So initially, the many research studies, they focus to identify who are that members. After that, they focus their study what exactly these bacterial members they are doing. And now most of the research they are working. After that? After their work, exactly, for example, when we take a certain medication for any particular clinical condition, how these microorganisms are reacting to these drug molecules. So that open a new path to identify new kind of drug metabolites, or even for new drug discovery, drug design based on the metabolites which can be generated by these microorganisms, can create a new way to identify the new drug molecules. So at the moment, most of the time, the area which I am working is to identify the microorganisms and their role in the metabolic route. So more soon we will focus towards what kind of role these microorganisms can have for diagnostics and as well as therapeutic purpose.
So one more question, sir.
Yeah.
In case of the antibiotic-associated diarrhea, will this, in case of like Clostridium difficile infections, can this microbiome therapy be promising to prevent such infections?
Yes, like for example, you mean like fecal transplantation?
The gut dysbiosis, which can occur due to either the antibiotics or anything, the Clostridium difficile infection, can that be prevented by using the microbiome therapy?
Yes, there are many clinical trials are going on based on they used to receive the gut microbial consortium from healthy donors, and they will transplant, or they will just give capsules which contains these microbial consortium, and they will try to regenerate the same healthy microbial environment. Though in those cases who had this Clostridium difficile infection, yes, in order to overcome this dysbiosis, yeah, there are many cases it has been reverted the pathologic conditions to healthy environment. But in certain, you have to be more cautious when you receive the consortium from healthy donor. Well, they have to be absolutely pathogen free. In some cases,
MNRCP04.
yeah, in some cases they got some infections, and in clinical trial there are some last year I think few deaths happened. So now they are again restructuring all the analysis to select the pattern, to select the how they can select the healthy donors. Yes, that's possible using microbiome based therapy. Yes, they can revert any diseases. Most of the research is ongoing.
Thank you, sir.
Good afternoon, sir. Good afternoon, sir. One more question related to it.
Yeah.
Can we use animal microbiota for the treatment of human being?
You mean animal microbiota?
For the human, in the treatment in humans?
Yes, why not? Once you isolate those particular microorganisms, yes, you can, once it is free from any pathogens, then yes, you can use any animal microbial transplant as also to treat human diseases. There are many, for example, from cows, human microorganisms, they are extracted as a consortium. There are many studies are ongoing as well.
Thank you, sir.
So now, Mr. Malik, you can ask your question.
Good afternoon, sir.
Yes, good afternoon.
So good afternoon, sir. I am Malik Maidul Islam. I am from MNR College of Pharmacy. Now I am pursuing Doctor of Pharmacy PharmD course. So I want to know what are the scope and opportunities for PharmD students in Qatar, sir.
So as I told earlier, PharmD, like what kind of job you are expecting? So mainly here, the opportunity currently which I see is you can be a chief of hospital pharmacist. More than that, here there is no much scope to be as a, because they here only recognize mostly the medical doctors. So the problem, even though if you have PharmD, here it can be recognized as a pharmacist.
Okay, sir. Sir, one more question, sir. For getting job in Qatar or any other countries, whatever exams are available for that exam purpose, is there any available books here other than our syllabus books?
It's normal. It's like same, like to be a normal hospital pharmacist, the basic questions, it's simple, like how you prepare for GPAT and other things, only based on, but in this mainly focusing on the medicines. You have to study more about the medicines, then there is no specific, there are many maybe training institutes in Chennai or any other, even Hyderabad, there should be many institutes. At the moment, I don't recall one. Once after this meeting, I can forward some institutes who can suggest this kind of training.
Okay, sir. Thank you, sir.
You're welcome.
Now, I am requesting Mr. Saran.
Hello, sir.
Yes, Saran, you can ask your questions.
Yes, sir.
Sir, I'm doing my B.Tech Pharmaceutical Technology now. What are the PG courses available which is best for me?
So, Jagan Mohan, do you want to answer this? I think your mic is muted.
So Jaganmohan sir, please unmute your mic and you can talk. Sir.
Yes. So, could you please repeat the question? So your question was, what are the opportunities for
What are the PG courses opportunities after B.Tech Pharmaceutical Technology?
Okay, so regarding the B.Tech Pharmaceutical Technology, so there is a university in Germany. It's called MLU. It is Martin Luther University. There you can pursue pharmaceutical biotechnology as a master's, which I have done by myself. So there is also a university, I think University of Hamburg, for which you need to send a pre-qualification check, where you can also do your PG in pharmaceutical biotechnology there in the Hamburg. And I remembered one more university, I think it's Aachen. It's ACHEN. It's called Technical University of Aachen, where you also have pharmaceutical biotechnology as a master's course. But always be aware that all the German universities do not provide English as a medium of instruction. So I think majority of the universities, they do teach in German, but there are also only few courses which you can get in English. So always be aware that before you apply, please do check the medium of instruction, which should be in English. Otherwise, you have to learn the German language up to B2. So there are 3 levels in German languages, B and C, which is A1, A2, B1, B2, and C1, C2. So you have to do at least B2 in order to get this admission. So which is quite hard, which takes at least 1 year to get this proficiency. So I suggest you to go for an English teaching course.
Yes, sir. What are the opportunities in India itself, sir?
Opportunities in India, you mean in terms of job or?
No, sir. After PG course, after pharmaceutical technology.
So especially in India, you mean, or in Germany?
Yes, India, sir.
To be frank, it's been like 10 years since I was looking for the job market in India. So I have really no idea how exactly it is now in India. But if I want to tell something in the place of Europe, so there is a really wide scope for biotechnology. And as you see, because of the new COVID thing, what happened since last 1 year, so the lot of money has been pumped into the biotechnology field for the research and also in the terms of startups. So there, I think there should be a lot of jobs that are coming up in a couple of years for sure. So
yes,
you can
sir.
you can go into quality control, you can go into the research development, or you can go into the molecular biology, basic discoveries of the targets, or go into the antibody purifications, or to do some antibody engineering. There's a lot of scope in biotechnology. I think in a couple of years, I think it should definitely boost a lot because of the recent pandemic.
Yes, sir. Thank you, sir.
You're welcome.
Now I am requesting Hima Bindu to ask her questions.
Yes, sir. Myself, I am Himabindu, pursuing my Form D 4th year. Sir, can I know what are the eligible courses that we can do expect in Germany, sir?
So you have done Form D, right?
Yes, sir.
So first of all, I want to clear you something. So Form D or Pharmacy doesn't make any difference in Europe. So you are still considered as a bachelor's.
Yes, sir.
Here also in Germany. And if you want to do something in the direction of clinicals or hospital pharmacy and this direction, you have to clear examination. It's called Startprüfung. So it's called, if I translate this, it's called a state examination, which is already again in German. So you have to learn German first as B2, and then
you have to give this examination in order to qualify yourself as a pharmacist in Germany. But apart from this, if you want to go into the other directions, I think you have to first broaden your how should I say? So you have to broaden your scope. Like you have to see to the in a bigger spectrum, and you can more specific directions, like going into a specific biotechnology or medicinal chemistry, or more into virology. So you have to go really into a specific directions. Then you can definitely, I think you have to go for a master's and then go for a PhD, because still your course is not considered as master's in Germany. It's still considered as a bachelor's, even though you have a PharmD.
But it is 6 years course now, sir.
Yeah, I know, but this
You
is
can
this is how
integrated PhD. You can do integrated PhD in USA or something. Now, a few universities have started some integrated PhD. It consists of master as well as PhD. So, in this area, you can join like masters integrated.
Sir, one more question, sir.
Yeah.
Sir, here in India, according to the guidelines, we cannot prescribe medicines directly to patients. So, is there any such scope in Germany, sir? Means we are having good knowledge about all the medicines now, sir.
you can definitely do a clinical pharmacy, but still the power to prescribe medicines is still holds by doctor. This is I think this is still the universal case wherever you go. In spite you heard a lot of news or hyping up that a pharmacy are prescribing drugs here and there. This is not true. This is absolutely not true. Still, the prescription powers are there with doctor. He is the one who prescribes, but the only thing what you could do is you can suggest. So for example, if doctor suggests something in the department of cardio, if he prescribes some nifedipine, maybe you can revert back him checking the patient case, and maybe you can say maybe nifedipine is the not right drug for him, and you can suggest. The suggestions are always welcome, but I think you have no power to really prescribe some drugs to patients.
So there is no opportunity for us, sir, in such cases. Okay, sir. Thank you. Thank you, sir.
No problem.
Our other researchers also joined. So on behalf of MNR College of Pharmacy, I wholeheartedly welcome Dr. Vishnu Nayak, and I am requesting Dr. Vishnu Nayak to give his opening remarks. Sir, it is not audible, sir. Vishnu, sir. Vishnu, sir. Sir, please unmute your mic, sir.
Now it is.
Now it is audible.
Yes,
sir.
yes. Now it is. Yes. First of all, thank you for all the participants and for joining in COVID situation. And the lecturer organized by international conference is very wonderful, because this more or covers like I am also working with the cancer and nanotechnology, so it's wonderful lecture. Thank you to everybody. And I'm as I'm listening, few people are raised some questions like how to check the natural compound structure, whether it is published or some natural compound structure as antiviral. So I suggest we have few softwares like SciFinder. We can put your structure, you can draw your structure, and you can search. So this it will give you all the details about your structure from the plant and everything. Then you can study your related article and these things. So we can make a conclusion that the structure is already reported, or what analogs are, what are the derivatives we can search. So nowadays SciFinder is very good tool. The another tool is Reaxis. The Reaxis shows how we can, because natural compounds are very complex in nature. It's very complex in nature and it's a big structure. So their total synthesis and those things, analogs, derivatives, and their total synthesis. This can be searched either SciFinder and then Reaxys. The another query that related to Turnitin. Turnitin actually it does not consider the chemical structure. It only considers the text, whatever the text you are adding. So I think this is the these things. We can only check the plagiarism in terms of text, not in terms of figure.
Thank you, sir.
So, thank you.
Now I am requesting Dr.
Then I would like to share some few good news. Something. What are the fellowships that are available to the Indian students in abroad? So we have nowadays we have for in various countries. If you consider USA, we have Fulbright Fellowship that is supported by United States India Education Foundation for masters, and then another one is Tata Scholarship that is supported by Cornell University, and from the UK we are having Flexi Fellowship. It is a very good fellowship, and from another one is Commonwealth Fellowship. We have to have more than 65%, and one English as a one subject. The Commonwealth Fellowship is very good. Then this Rhodes Scholarship. Then the another fellowship is from the Germany, DAD Scholarship. So this is good, and a few students recently joined with Mary Curie, and in Israel special fellowship is a Lady Davis Fellowship. The another thing is, instead of other than this fellowship, we have if you approach the professor, is there any university professor? So if the professor accepts your credential, so university because professor, he will approach to the grad school, because in foreign every university they are having grad school. So grad school, they will fund to the students on behalf of professor. So this is very good fellowship, because in other fellowship like that, there are a lot of competitions and those things. A lot of competition, they will receive the application from the throughout world. But in grad school, the professor selects the only students, those are those who have experience in their related field. Their field only. If professor working with cancer, if professor working with the in cancer also, we are having many because synthesis, biology, and nanotechnology, like the molecular modeling. So if the professor is agree, then he approached to the grad school along with the student's profile. So this is good opportunity if you can mail to the professor if he accept. So means there will be more chance to get this fellowship through grad school. So for that, you have to have some GATE and GRE, IELTS. They accept this course. So grad is, in my opinion, grad school is fellowship is best. The other, because other fellowship are also available, but if you are interested in particular topic, so if you are interested in particular topic and you are interested to do under in expertise way, so grad school fellowship is good. So it's your choice. So there are the fellowships.
Thank you, sir.
Thank you.
Dr. Faniti Pika to ask her question.
Good afternoon, sir. Myself, Dr. Sanidipika, sir, Biotechnology Department. My question to Selva Shankar, sir. Sir.
Doctor, yeah. I look forward to your question.
Sir, if we want to opt a faculty job in any of the UAE countries, this is from long back I have the doubt, sir. So what is the procedure for applying, sir? After doctorate, after getting funding and everything, if we have a fund in our hand also, we have completed a project. Is it safe for women to continue as a faculty in any of the UAE countries, sir? As a faculty in life science universities or medicine universities like that?
What do you mean by the safety? You mean
yeah, it is a UAE countries, no, sir. Is it okay for ladies? I'm asking.
Yeah, there is absolutely no problem for ladies here.
In all the Oman, Qatar, everything, sir.
Everyone actually here, it's one of the safest country. All most of the Arab countries, they are more safe to for women. And since I'm in Qatar for almost 4 years, it is you can go at any time in the night time. Even ladies also, you can. There is absolutely no problem to find a job directly in universities. Once there is any announcement for the vacancies, based on your number of publications and your teaching experience, after revising your profile, yes, they will contact you. But here the process is little slow. Once they made the announcement, they take almost 1 year to process all the applications, and they will call you for the interview. So you have to wait for with a little more patience.
Okay, sir. Okay, thank you, sir.
Welcome.
There is one general questions in the chat box. Sir, why there are various indexing and abstracting is available for journals?
I think Dr. Vishnu Nayak or Dr. Sanjay Sangar, anyone can answer.
Okay, I think Vishnu is going to answer. Yeah, after you.
Actually, in India, we are considering like UGC listed general like that, but in SCI indexed. But in abroad, they will consider quartile. Like you can search, you can put SCI Mago, SCI Mago. You can search your put your general name, and then you can search. In abroad, they will mostly consider quartile 1 and quartile 2. So in India, even if you have a low impact factor general also we will consider, but in abroad, to get fellowship or to avoid the PhD, they don't consider about the impact factor. They consider quartile 1 and quartile 2 like that. So because I came to know, like World Journal of Pharmacy, they are in quartile 4, quartile 5, but their impact factor is 6, 7 like that. So sometimes we are misinterpret what is going on. 6, 7, 10 impact factor. So you can search SCI Mago, then there you have quartile 1, quartile 2. In abroad countries, they will consider quartile, not the index something. But some that is depending to some university to university. But nowadays everybody is following the SCI Mago, their ranking.
Thank you, sir.
I think.
So just to add on to the Dr. Vishnu Nayak sir's previous quote, like regarding the grad school scholarships. So the major difference between the other scholarships to the grad school is grad school usually has a kind of a curriculum. So for example, if you take in case of Germany, for example, so in order to get your PhD, you have to at least get 180 credit points in the university to defend your PhD. So grad schools usually have a curriculum which is kind of like a syllabus where you have to definitely participate in the presentations, do some oral presentations, go for lectures, and do some inter and intra lab works and journal clubs. So you are kind of forced to do this and to get the credit points. So this is also ensures that your PhD is done on time, and also your funds are secured. So once you are there in the grad school, then you have multiple opportunities. Also go to the other abroad labs to do some kind of a short 2 or 3 months trainings, which are also funded by the grad school to you, so where you don't have to spend it from your hands. And at the same time, so this is something which is not happens usually, but if you also have any problem with your supervisor, sometimes if your supervisor is not good at some point of view and it's kind of making you going crazy in your work, then you can also approach grad school to solve the issue. So you always have a kind of a helping hand behind you, that if you are joined in a grad school, then this is kind of a secure to finish your work.
Thank you, sir. There is another general questions that any one of you can answer this one. They are asking how could we select the project for our PhD? On what basis we can select our project? Which topic we can do like that?
So maybe I can start with my experience. So especially in Mexico, it's not like mainly once you join with certain scientist after your his field of interest, right? So in most of the scientists in Mexico, already they might have some project which is already funded. So they will propose you to work on that particular project. If you want on your own novel idea, in that case, yeah, then who will fund you? Then in that case, it's not possible. That can be possible during your PhD time. Then you can work with your scientists and you can write a grant. Then that can be funded. If once it is funded, then you can continue with that project as your postdoc experience. I did in my career like that. In the same institute, I continued as a postdoc fellow.
So
Others, they can share their experience.
Just to add on to the Selva Shankar sir, as is rightly said. So first of all, what is exactly you have to think is what is PhD. So you have to have in your mind what exactly is PhD. PhD, if I define PhD in my words, it's like questioning your own question, answering it by yourself. So you will question your own question, answer it by yourself. So this is exactly what PhD is. So in order to do a PhD, the first thing is not which field you want to go. First, you have to have the right motivation, because you'll be staying in this field for at least 4 or 5 years in order to finish these PhDs. And if you have a novel idea, for sure you can approach in the same field to a certain professor, that you can send a proposal to him that you have some kind of a novel idea which you want to pursue. Then if they are also interested, then they can divert some funds from their own group and then take you into their group. And because every researcher also wants to have novel ideas, so which also brings new papers and also get more funds for them. So this is also an add-on things to them. So if you have some novel ideas, it's always encouraged you to apply or send a kind of a motivation letter to them that I'm having this idea and to do it. But at the same time, in majority of the labs, usually the projects are not really 1 or 2 pages from, it's a kind of a very big project which is kind of divided into multiple small sub projects, and these sub projects are given to many different researchers, and then they're funded equally in these directions. And as Selva Shankar sir already said, so it's already an ongoing project which is there, which has to be answered in a multiple directions and also have multiple questions to be solved. So you will be hired kind of a job, and you can do your PhD to answer the questions if you are interested in the same field. But as a student, I was also a student from MNR in 2011, and I absolutely understand the current situation, how you have. So as soon as you finish your bachelor's and you think your mindset is really kind of really bit narrow now. So first of all, I want to tell you that you have to first broaden your scope. So you have to check the broadness of the pharmacy, the field. So this is really an interdisciplinary field. I think you give examination of 50 subjects, so you can go into each of the 56. So each subject is kind of a one complete whole new world. So if you're really interested in these directions, then to really think what you have to do, which direction you have to go, and then you can pursue PhD in these directions.
Thank you, sir. So
then I would like to tell a small thing to all the participants. If you want to pursue a good PhD or something, or if you want to select a professor or something, if you want to apply, just the funding is available in two terms. One is hot topic. Currently, the one is hot topic, and then is another one is neglected disease. One is hot topic means like corona and cancer, central nervous system, and the another neglected disease. Neglected disease means we can neglect. Just a few diseases we can neglect. So you chose your topic like a hot topic, means that is beneficial. So just to see the mortality, how many people are affected with this disease. So when you are choosing the topic, this is also matters, because you need your funding for your PhD, 4 years, 5 years. So just select the hot topic. And that is good way. And the funding agency think, because we received a funding agency from Thailand Research Council, and now we are working with Bio-Pharmaceutical. So they will consider how your research is useful to the society. So there are two. One is commercial, and then another one is some non-commercial. Because if you synthesize, if you are doing with some central nervous system, because some depression or those things, those are some disease can be modified or can be cured by counseling or those things, counseling and some patients this thing, a few minor things. But some disease you cannot it is necessary to take a medicine or those things. So this will go, this consider as a commercial. So I think if you can go in the commercial way, so just because many universities are many funding agencies are supported, because they will think in terms of money, how much money we are investing and how much money we are getting back. And so just choosing the hot topic, and choose your topic and just mail to the same professor who is working in the same area. That will improve your chance of acceptance. Because blindly sending mails to different professor, even if it is unrelevant to your area, they will not see your profile. Just work on it. What are the hot topics, which disease we have to choose, and meanwhile select the publication of the professor, same area, and that will be helpful to get the funding in short time.
thank you, sir. Now I am requesting Mrs. Sai Jyoti to ask her questions.
Good afternoon to all. Myself Sai Jyoti. So my question is, what are the women fellowships is available to do PhD in India or in abroad?
So maybe I can start in the consent of Europe. So here, for example, I would just want to tell like a short analogy. If a two applications comes to a professor who have almost equal curriculum and have done almost equal weightage, if you are a woman and if you are a man, if this guy is a man, then the woman gets the first option. She gets the job.
So here, I think since 15 years, I guess. So they want to maintain more female researchers in the field of science. So they want to make a ratio of 40 to 60. Now it is around 40 to 60, which means the woman to men ratio. So they need a 60% of females to 40%. Now ratio is around 30 to 70 here. So there is a huge scope. So if you are a female researcher, then the chances of getting a PhD or getting a job or getting admission is comparatively high.
Thank you very much.
So I think someone asked in the comments, is there any scope for jobs for women in Arab countries, especially in clinical trial organizations and pharmaceutical production industries? There is no particular discrimination in Arab countries. Those who have the real eligibility criteria, of course, they will hire you. But at the moment, in last few years only, Arab countries, they mainly started to focus more towards the research. So still they are in the preliminary stage. So still they don't have a particular industries focusing on pharmaceutical or the clinical trial. They just started. Maybe in after next 5 years, definitely they will have huge opportunities. So during that time, definitely they will recruit the right eligible candidates. There is no particular, they depends upon the eligibility, they will select. For example, in our department, most of the leading scientists are female. So there is no particular discrimination in this. Thank you.
Good afternoon, sir. Myself, Sandhya, working as assistant professor in MNR COP, sir. So I have a question like, based on experience in India, can we get a job in abroad? Like without writing any exams or anything, like through public relations? Is there any possibility? Have you seen any examples of anyone who is doing job there based on experience in India? Either in
Yes.
research or
So you mean you want to work in any university here in Arab country?
anything, any job opportunity, but with
Yeah, except from to be a pharmacist or other thing, you can directly apply for, it depends upon the job vacancies. Yes, you are eligible to apply. So to be directly, for example, in academic area, they will expect you to have the teaching experience and your research experience. Based on that, they will hire you. There is no particular exams for that. Just you have to clear, you have to be show your talent in their interview.
So if we need to work as a pharmacist there, so the experience in India, will it calculate or again we need to attempt exams to qualify there?
Yes. Anyway, you have to, your experience will be counted, but still you have to clear the exam. Unless you clear your exam, you cannot work as a pharmacist here.
Thank you, sir.
You're welcome. I think Jahan Mohan is want to tell something.
Sorry.
Yeah. So just to add on to the statements what Sir said, rightly said. So first of all, your experience always counts. Your experience always counts, even though you do it in India, you do it abroad, wherever you do, the experience always counts. But during the interview, as Sir said, you have to prove yourself that you really did this. So there is also a kind of a wrong notion that comes up in some of the foreign countries, especially in US and Canada, that if you have a work experience from India, then you are subjected to a trial. So they really doubt that you really did this because of the bureaucracy and the ease things what happens within our country. So there is a this short trial that people wants to test really that you really did this. But if you really have some experience, the experience always counts. And in terms of, not in terms of pharmacy, but if you want to go for a production or if you want to go into industries for research, definitely your experience 100% counts. It also happened in my case. So I also got a job as soon as I finished my master's. I did job for 2 years in the biotech, and in order to get this job, I actually did a 7 months internship at Aurobindo, so which really helped me to get into this job. So they asked few questions in terms of the HPLCs and some viral productions, which I could answer because of this internship. So this really helped me to get into the job. So there were also many other German guys who were more proficient in also in German language, but still I got the job because I had this experience. So experience always counts, but it has to be genuine and true.
Thank you, sir.
Saravanan sir, it's my turn. So I just want to ask one question related to journals. So many of research, I mean, doing PhD as well as M.Pharm, they're unable to get some subscribed journals. Normal journals they may get. So is there any website where those people will get subscribed journals freely? If you help, I mean, if you give some websites, so all PhD scholars will be very happy.
So I think I should not tell this, but almost everyone who is in the research field, they are using this. Might be everyone might have heard about Sci-Hub.
Like Science Direct, Bentham Sciences, so many of the journals we need articles from different journals. Is there any source?
So Jagan Mogan, Vishnu, you guys want to tell something?
Actually, yes. SciFinder is a good source, but nowadays we came to know they are also going to close, and a lot of debate is going on SciFinder. But now academia, and then another one is ResearchGate. In those you can find full text, because whatever paper is published, people are just uploading their credential in the site. So I think ResearchGate is now very helpful. Some universities or some colleges nowadays, and I think moreover you can make the friends, those who are in good university. So in good university they have all access, so they can just, that is just one click, 1 minute work. To send a research paper is not going to take your lot of hours and hours. So make good friends, and that will always help. They have access for all the publications. So I think friends can help in this regard, I'm thinking.
So only friends can help. We cannot download it from
Actually, to add on both, sir, the thing is, there is also some soft spots that you could really try. So that you can make some LinkedIn connections with these guys, and actually you can also write, and you can just ask him that, could you please send your recent publication. Actually what happens is every scientist is kind of enthusiastic that if other scientists read their paper, they're kind of really happy for this. So if you directly ask them to send the paper, they will do it. This is a soft spot which you can trade, but as Vishnu Naik sir said, it's really a one-click thing for especially students in abroad, that they have access all the time. I also have access to almost all the major journals, which is kind of free for my institute. So if you have any journals that you need, please let me know. I can send you. It's not a problem.
Definitely, I will ask you. Don't
Definitely.
Thank you.
Another thing is, in the publication full text, we have the email address of the corresponding author. So if you can mail to the corresponding author, definitely they will respond you.
Yeah.
Because if you study their paper, and if it is good, you cite their publication, so they will also get, indirectly they will also get the citation. So just mail to the corresponding author, so that will help you.
No, actually, I mailed only few members. They sent their article.
Yeah.
So many of them they didn't respond. Okay. Anyhow, thank you.
One more thing is, actually, when you send emails from your personal email IDs.
Okay.
So if you send it from the personal email ID, majority of the emails are directly sent into junks or into spams.
Okay.
is one of the major problems in all the abroad countries, that all the email IDs, what they usually cite on the papers, they are from the institute. So if you send it from an official email ID, from the institutional email ID, so they will directly go into the inboxes to check the emails. So this is one of the problems usually every Indian student has, especially not privileged university students have this problem. So I also faced these issues when I was there in India, because there are some universities in Canada, I guess, I think it's McGill and Toronto. If you want to apply to these universities, you have to apply through your own student email ID, which I did not had. And I don't know when now also if student from MNR has this. So I also request Professor Algar Swami sir, if this could be one of the thing that you can provide to the students, this will definitely help them to get this identity via email. So it looks more genuine also in the application, and this helps the students a lot. So I really faced this problem for at least couple of years. So maybe this would be a nice add-on to the students if they get this web mail from MNR. This would be really nice.
Thank you, sir. Thank you, all of you. And even though we are getting questions from YouTube's also, but due to time schedules that we are closing this session. So now I am requesting our principal sir to conclude these interactive sessions.
So thank you all the participants and the resource persons, Dr. Vishnu Nayak, Dr. Selvu Shankar, and Mr. Jagan Mogan. So when we have planned this live interaction with the international researchers, our students, that is all three are my students, they have accepted our request without any hesitation. The reason why we have selected these speaker is not that they are my students. We have selected them because they are all now upgoing or developing scientists. So now they know each and every practical problem of the current students. That many of the students of undergraduate or postgraduates who are doing and facing the problems, so they can address well. So rather than calling any senior scientist or expert scientist, the students are the growing scientists, if we call, it will be very much useful to the our fraternity. So with this mindset only I approach. They also immediately accepted our invitation. That shows the affection they are showing to our teaching as well as to the college. So really we thank all of them for accepting our invitation and sparing their valuable time with us. And really what we'll do, if at all some more questions comes, we'll have one more interactive session. So you people should, like what we have done for our students, you people should do something for our budding pharmacists and scientists, so they feel happy and they feel getting this kind of guidance is not possible by others. So really in the coming days, if I get more queries, I'll contact all three of you, and you please will plan one more session for this type of live interaction, so that we can feel happy, many of our juniors. So thank you all. Kindly accept our small mementos, virtual mementos. We are prepared for you in order to appreciate. Hope you will feel extremely happy to give this memento, because all of you, you know, during when you are doing B pharmacy, we give during our first year, second year, whenever you take first mark, second mark, we give prizes. This is for your research achievements. I am on behalf of MNR College of Pharmacy, the management, and all our students and all delegates, we are giving this mementos. Kindly accept it, and thank you very much. Thank you all.
Thank you very much, sir.
Thank you.
Thank you for your wonderful work, sir.
Kindly accept it and save it as a soft copy for your future purpose. Mr. Jagan Mogan, Dr. Selva Shankar, and Dr. Vishnu Nayak. Thank you. Thank you all. We'll go for the valedictory. As we are running short of time, we'll meet some other time. Thank you.
Okay.
Thank you, sir.
Thank you, sir. Thank you all. Now I invite Dr. Vishaka Kulkarni, Madam, Associate Professor, Department of Pharmacognosy, to conduct the valedictory ceremony. Please, ma'am.
Thank you, ma'am. A very good afternoon. And on behalf of Nanar College of Pharmacy, I very warm welcome to all of you to this valedictory function of International E-Conference on Recent Developments in Biopharmaceutical Sciences. Since morning, we have had deep insights into biopharmaceutical concepts. The researchers from different countries have enlightened us by sharing the current trends in biopharmaceutical sciences. The live discussion was really helped to participants, delegates, and the faculty also really in clearing our doubts regarding education, regarding publication, regarding the scientific doubts. Everything has been cleared during the live discussion. It was excellent session. Now we are at the end of the program, end of our today's e-conference, that is recent developments in biopharmaceutical sciences. So today we have with us Professor Kavita Vagre ma'am as a guest of honor. She is the head Department of Pharmacy, Usmania University, and throughout our MNR College of Pharmacy journey, Usmania University always supported us and helped us for the development and growth of the college. Kavita Vagre madam, especially she has supported us during our every event. I welcome you on behalf of MNR College of Pharmacy, ma'am. I request ma'am to have a valedictory remark. Kindly address the gathering, ma'am. Kavita Vagre ma'am, welcome and kindly address the gathering, please.
I really thank everyone and the organizers for inviting me to this international e-conference on recent developments in biopharmaceutical sciences. And I think MNR College is the first one to organize such type of a conference for the students where we are having live interaction with all the experts, clearing all the doubts what we have when we are doing research. I appreciate the management and the principal and all the faculty members for organizing such an event, because they must have gone through they must have experienced what problems they are facing when they are conducting research. So it is really very nice, and all the students are very fortunate to be part of this webinar. I thank the organizers for conducting such program, and I feel that in future also you can conduct such programs with experts who are there, like Dr. Selva Shankar, Dr. Vishnu Nayak, and Dr. Jagmohan. I really thank you all, your patience and come joining the seminar and encouraging our students and clarifying their doubts. Thank you very much, sir. And I also thank the management staff of MNR College for inviting me for this program. Thank you very much, sir.
Thank you very much, ma'am. Thank you very much. Your presence really graced our valedictory function. Thank you for accepting the invitation. Now I request Dr. Vinyas, Associate Professor, Department of Pharmacology, to give a vote of thanks. Vinyas, sir, please.
Thank you. Good evening, everyone. Myself, Dr. Vinyas, Associate Professor, Department of Pharmacology, and I'm here to present the vote of thanks for today's webinar. I would speakers, Dr. Shashank from the Stanford University on the nanotechnology in the new dimensions for stem cells therapy and diagnosis. I'm pretty sure that precious knowledge that Dr. Shashank gave us will definitely help us in our studies and future. And the next speaker is Dr. Carlos from the Mexico, taking time from their busy schedule and gave a wonderful lecture on the nifedipine and omeprazole on pharmacokinetics. And he has enlightened us the knowledge with the pharmacokinetic parameters. Thank you so much, sir. And I would also thank Dr. Janet from Spain. Angeles, she has given an excellent topic on the clinical and therapeutics use of biomarkers in the breast cancer. Today, we experienced a novel insights in the biomarkers in the breast cancer. Her knowledge on the oncotype diagnosis was profoundly interesting. Her research tools, inside hybridizations and multiple gene expression for the pathogenesis of cancer was excellent.
And the session 4 was with the live interaction with the international speakers, Dr. M. Selvashankar sir, Dr. Vishnu Naik sir, and Dr. Jagan Mohan Germany. They really helped the researchers and the students, and they clarified much of their doubts. And I would finally I would like to thank Principal Dr. Algar Swami sir for providing a unique platform and inviting international researchers and speakers to conduct a unique webinar. And I would also thank management, MNR Education Trust, organizing committee who have really worked hard to make the event successful. Also, I would like to thank all the students present here for paying their attention and learning. I would end up my speech here. Thank you, one and all.
Thank you, sir. Thank you for your vote of thanks. Now I request Dr. V. Algar Sami, sir, Principal, MNR College of Pharmacy, to as a token of love and appreciation, to provide a virtual memento to Professor Kavita Vagre, ma'am. Ma'am, kindly accept the virtual memento.
I request Dr. Saravanan to. So Dr. Kavita madam is always support to us, all the faculty members and our college growth. Recently we have received the, our college has been recognized as Center for Research Center, and it is one of the very big remark. And many of our faculty members also have been recognized as PhD supervisors. All this growth has been happened with the support of the University College of Technology. And madam is very much supportive to us. In order to appreciate the madam support and today's August presence, we provide this virtual memento to Professor Kavita Vagre. Kindly accept it, madam, as a token of our appreciation and love and towards your that is professionalism. Kindly accept it, madam.
Thank you so much, sir. I really appreciate the way you are conducting the conferences. I am very happy, sir. Still I need you to do more and more, because science is so vast. It's never ending. Please do conduct many more webinars to enlighten all our students.
Definitely we'll do it, madam, and we'll prove our MNR College of Pharmacy and the Osmania University affiliated colleges in the significant part in the profession of pharmacy.
Definitely, sir. We will be at the back of you. We will be supporting you, sir.
Okay. Thank you, madam. Thanks a lot.
Thank you, sir. Thank you, ma'am. Now, at the end of the session, I request Saravanan sir to play the national anthem. Sir, kindly play the national anthem, sir.
जय हे भारत भाग्य विधाता, पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा, विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा, तव शुभ नामे जागे, तव शुभ आशीष मांगे, गाहे तव जय गाथा, जन गण मंगल दायक जय हे भारत भाग्य विधाता, जय हे जय हे जय हे, जय 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 हे।
Jai Hind.
Jai Hind. Thank you, one and all. Herewith I conclude the session. Saravanan, sir, please conclude the session.
Thank you, one and all. All delegates are requested to provide their feedback. So already we sent out a feedback link in that your email ID. So please provide your valuable feedback, and within 3 days you will get your e-certificate. All the participants will get an e-certificate through your registered mail. So please provide your valuable feedback link. So we will once again we will meet in that another occasion very shortly.
Thank you very much. Thank you all. Bye bye.
Principal sir, can I end the meeting?
Yes, sir. Thank you very much. Thank you all, madam. Kavita, madam.
Yes. Thank you so much, sir.
Your granddaughter also joining.
Yes, sir. My granddaughter also joined.
Okay, okay. Thank you, madam. Nice, madam. So sweet.
Can I say something
Jagan Mogan,
about the corresponding?
Jagan Mogan and Vishnu Naik and Selva Shankar.
Yes, yes, sir.
Yes, sir. Thank you all.
Thank you so much for inviting.
Okay, bye.
Thank you all. I'm ending the meeting, sir.
Okay, sir. Log